Hello, Nita.
Good morning, everybody.
Good morning, sir.
Good morning.
Sir, good morning, Sharma sir. How are you?
Good morning, sir. Good morning, Professor Rajgopala. How are you, sir?
Sharma sir, good morning. How are you?
Good morning, sir. Good morning. How are you,
Good.
Professor Rajgopala?
Yeah, I'm doing good.
Yeah.
So,
Okay.
how
Great.
are you doing?
I'm fine, and thanks for accepting our invitation. Thank you very
Oh,
much.
thank you. Thank you so much. Thank you.
Yeah.
Good morning, sir.
Good morning, all of you. Good
Yeah,
morning.
good morning, sir. And good morning, Professor Sharma.
Yeah. Good morning. Good morning. So this is actually our annual exercise, just to inform you. And
Okay.
since we are UGC SAP department in third phase, though the program is over now, and we will be approaching the UGC when they start with this program again. So now we have made it as a national, our annual exercise, and we were lucky year before last that we could organize this program on physical made, and then the COVID came. February we organized, in March the COVID came.
So
And,
actually, last time, the SAP committee came to our department, they also said the same thing. Why don't you conduct a national seminar?
Okay.
Right.
But we have conducted one national seminar where, same thing like a webinar last year.
Yeah. Yeah. So we have been very regular in this exercise because it helps our affiliated colleges. And in this university we have about 8 colleges which offer undergraduation in botany.
So how is the situation in Goa? COVID situation?
It's good for visiting, no problem.
Going down? Yeah.
Yeah, it's going down.
It's coming down, sir. Yes.
Hyderabad is worst actually.
Okay.
So almost everyone affected.
Yeah, but I think it is not going to last very long. It is going.
Yeah,
It's not very severe anyway.
So the program will start at 10 o'clock, right?
Yes, sir. Yeah, it will start at 10. Yes, sir.
So we have a very young colleague who is organizing this
Dr.
webinar, Dr. Jalmi.
Oh, Jalmi, yeah.
Yes, and she did her PhD from that NPPGR, New Delhi.
NIPGR.
NIPGR, yes.
Okay. They are one of the best now in the country,
Yes.
They are really doing good.
And she's also good, actually.
So how many are there now in the botany department? How many faculties are there?
We have 8 positions, but 7 are filled. One is still vacant.
Okay.
Yeah.
But still less.
We are a small department. It's just, because our strength is also very small. the Goa has a population of 12 lakh people. You can understand, we get about 10,000 students appear, 10, 15,000 appear for 12th standard. And then we have so many professional courses, engineering and computer science and
I can understand.
medical science and all that thing. But if you are left for taking such kind of courses.
Good morning, sir. Professor Bhatt.
Good morning, Chris.
Yeah, good morning. Sir. Nice to see you.
Nice, yeah. It's my pleasure, yeah.
अभी मैंने अपना वो ऑन नहीं किया कैमरा so I can talk without
Good morning, sir. Professor Alok. Good morning, sir.
Good morning. Good morning.
Yeah. I'm requesting Dr. Siddhi to put for recording. We'll start the event.
Yes, sir. The recording has started.
Good morning, everyone. Honorable Vice Chancellor, sir, deans of various faculties and schools, heads of various departments, colleagues, resource persons, students, and ladies and gentlemen. I'm Professor S. Krishnan, Head Department of Botany and convener of this webinar. I have the privilege to welcome you all for this national webinar on advances in plant sciences. First and foremost, I welcome the Honorable Vice Chancellor of Goa University, Professor H.B. Menon, for this inaugural function, whose dynamic leadership I'm sure the university will grow into further greater heights. Welcome you, sir, for this inaugural function.
Thank you.
And thank you for sparing your valuable time with us and gracing this occasion and kind acceptance to release the abstract book and to deliver the inaugural address. Also, I welcome Professor Vijaya Kerkar, Dean, Faculty of Life Sciences and Environment, and all my colleagues, Professor Rodrigues, Professor Sharma, Professor M.K. Janardhanam, Dr. Rupali Bandari, and Dr. Siddhi Jalmi, who is the organizing secretary for this national webinar. I take this opportunity to welcome Professor DJ Bhatt and Dr. Nandkumar Kamath, former colleagues,
Yeah,
who could join on this occasion. Welcome you, sir.
thank you. Well,
Special welcome to all the resource persons from various reputed institutions from India and abroad, and thank them for their kind acceptance to deliver the invited talk. Also, I welcome the faculty members across the university, affiliated colleges, and participants across the country from various universities and institutions. Also, welcome all the research scholars and students across the university and outside the state. Originally, we have planned for 2 days offline national seminar. However, due to the increasing number of COVID cases, we have changed to the webinar mode. I'm happy to inform you that 309 participants have been registered for this webinar. Since we have more than 300 registered participants, we have provided the Google Meet link only to the resource persons, faculty colleagues, and some research scholars. And this webinar is broadcasted on Goa University YouTube channel for students and other research scholars and participants.
Okay.
On this occasion, I would like to present a very brief account of the department. The department was established in June 1990. Making a modest and humble beginning in a small space, the Department of Botany has transformed itself over the period of past 30 years. It has now created a strong base in teaching and research, mainly because of wide range of specialization by the faculty members on the subject, such as plant diversity, algae, fungi, bryophytes, pteridophytes, and higher plants, systematics of angiosperms, endemic flora of Western Ghats, mycology, molecular plant stress physiology, climate change, and crop productivity, bioprospecting, plant physiology, plant biotechnology, plant tissue culture, molecular biology and genetic engineering, mycorrhizal research, plant developmental biology, plant histochemistry, rice grain biology, natural dyes, nanotechnology, phytochemistry, algal physiology, biochemistry, plant microbe interactions, and plant cell signaling. The department has excellent herbaria of higher plants, and we also have Goa University fungus culture collection, plant tissue culture laboratory, and polyhouse, and a botanical garden for ex situ conservation of plants of Western Ghats regions. The contributions of all the faculty members of the department, starting from Professor late S.G. Thorne, Professor S.R. Yadav, Professor Bhatt, and Dr. Nandkumar Kamath, and all the present faculty members, Professor Bernard Rodrigues, Professor P.K. Sharma, Professor M.K. Janardhanam, Professor Vijaya Kerkar, Dr. Rupali Bandari, and Dr. Siddhi Jalmi is highly recognized and also appreciated. I'm happy to share that a department is so far produced 60 PhD students. Presently, 25 students are pursuing for PhD. Over the period, the PG student strength has increased from 10 to 38. The alumni of department are well placed within the state and in other parts of the country, and in countries like Canada, USA, New Zealand, UAE, and other countries. The department is now known around the nation and elsewhere as a center for studies in modern botany, with first-class teaching and research facilities available for young students. More details about the department and Goa University is provided in the abstract book. In this webinar, we have 10 eminent resource persons, and among them 2 from abroad, and they will be sharing their experiences, covering a wide range of topics, and I'm sure we all will enriched from their knowledge. Once again, I welcome you all for this webinar. Thank you. Now I request Professor Vijaya Kerkar, Dean, Faculty of Life Science and Environment, to give her introductory remarks.
Thank you, Professor Krishnan. Honorable Vice Chancellor, Sir, Professor H.B. Menon, eminent resource persons, Professor S. Krishnan, Head Department of Botany, Dr. Siddhi Jalmi, the Organizing Secretary, and all the participants of National Webinar on Advances in Plant Sciences, and students from the various departments, warm welcome and best greetings to you all. At the outset, I congratulate all those who are involved in organizing this event, and I'm sure with their experience, dedication, commitment, and zeal, this webinar will be a grand success. Although I am addressing as a dean today, I'm very proud to say that I am a professor from the Department of Botany, Goa University. It is said that Utsavena hi mandiram. This Jnana Mandir, Department of Botany, Goa University, has been celebrating several Utsav every year, organizing conferences, workshops, seminars, and symposia, basically to create awareness regarding the research in the various aspects of plant sciences. Today's webinar too is encompassing many disciplines and applications of plant sciences. I'm sure this webinar will provide a platform for every participant to gain the knowledge from scholarly eminent personalities, and the deliberations will be of great benefit to each one of you. It would have been really nice if the seminar was offline. Goa being a charming pocket of biodiversity, biodiversity-rich Western Ghats would have added grace for the scientific and academic discussion. Once again, I congratulate the organizers for their foresight in organizing this webinar and wish the event grand success. Thank you, one and all. Thank you.
Thank you, madam. Thank you. Now I request the Honorable Vice Chancellor to release the abstract book and deliver the inaugural address. Sir, please.
All the 3 of us would stand and then show that, or how it is?
Yes, sir. Yes, sir. Yes, sir. We can do that.
Okay.
Thank you, sir.
Thank you.
Thank you, sir.
Am I audible?
Yes, sir. We can.
Yes, sir.
It's very clear, sir.
Professor Vijaya Kerkar,
Yes, sir.
Faculty of yes.
Yes, sir. We can hear you, sir. Clear, sir. Yeah,
Okay.
clear.
Okay. Professor Vijaya Kerkar, Dean, Faculty of Life Science and Environment. Professor Krishnan, Head Department of Botany and convener for today's webinar. Dr. Siddhi Jalmi, Organizing Secretary for today's webinar. My dear colleagues from Department of Botany and across various schools of Goa University and across the country, eminent resource persons from across the country, and participants, ladies and gentlemen. In fact, I am really delighted and extremely happy that Department of Botany is organizing a webinar on advance in plant sciences to keep abreast or update the new developments in this branch of knowledge in the midst of this pandemic. In fact, the original intention was to hold an offline meet, but with the prevailing condition prevailing in Goa, it was ultimately decided to go ahead with the online meet. My congratulations to all those instrumental in doing so, especially the convener, Professor Krishnan, and the organizing secretary, Dr. Siddhi. The two biggest challenges that humans are facing in the 21st century are food security and deteriorating environmental health. Plants are not only food producers, but also an essential part of the clean environment. Modern agriculture provides the raw materials for most of the world's food and for many industrial products, including textiles. It plays a critical role in human society, serving as the backbone of the economic system. Significant scientific and technological advances over the years have led to great increase in agricultural productivity. Nevertheless, agriculture systems of the future face huge challenges in balancing and optimizing productivity and profitability against the stewardship of ecosystems and natural resources. Therefore, there is a great need to invest in new research and technology to transform plant biology for human needs. In fact, the webinar on plant science today will cover all the essential topics, extending from response of photosynthesis to abiotic stress, plant secondary metabolism, biodiversity, and taxonomy, along with the medicinal plants. I'm sure that this online meeting will offer an exclusive platform to talk over the research developments and major challenges in plant science and research. Surely this will also provide an online open forum for students, young researchers who are in their initial phase of career to listen and discuss the challenges and share their latest research achievements in this knowledge domain. In fact, I'm extremely happy to note that the webinar starts with the topic, response of photosynthetic apparatus towards abiotic stress. This is very pertinent in the present context. If you look at the degree of atmospheric pollution going on in India, it's alarming and shows an upward trend. Therefore, plant scientists need to look at the pollution stress on plants. A glaring example is burning of crop residue by the farmers and the resultant pollution in capital city of Delhi, particularly in November, December, when the air density increases. Since the air density is high, it traps the aerosol, which means that the residue, as a result of burning of crop residue, what is being emitted to the atmosphere is soot particles, or which we scientifically we call that as black carbon, BC, and that is being emitted to the atmosphere as a result of this burning, and thus increases the concentration in the atmosphere. These being fine mode particles, it easily covers the entire leaf, thus affecting the rate of photosynthesis. One aspect is that it's the winter season, therefore PAR, photosynthetically available radiations, are less, and another aspect is that PAR gets diffused in the atmosphere, which is heavily loaded with the anthropogenically derived particles. And third is further reduction of this PAR due to settlement of BC or these particles on the plant leaves. This in turn decreases the ability of carbon dioxide sequestration by the plants and thus augments the atmospheric carbon dioxide, and thus the increase the atmospheric temperature and of course the global climate. Another effect is the direct one, whereby BC means this black carbon aerosols of the particle absorbs incoming solar radiation, especially the PAR radiation, and increases the ambient atmospheric temperature and thus the climate. In fact, BC, if you look at the scientific articles, which clearly shows that BC is second to carbon dioxide as far as the absorption of insolation is concerned. Therefore, this is an important area of research, and this can be put in terms of climate change, rice production, impact, and adaptation. To tackle such a problem, one needs to look at a coupled numerical model so that you can even come out with a prediction model. For this, three tools have to be used. One is in situ data, that means the data you collect from, the ground truth data or from the area of interest, you're directly collecting using the different instruments. And second one is the satellite data. And the third one is the numerical model analysis. So with these few remarks, wish you all the very best and let you have some fruitful discussion during the coming 2 days and let it throw a trajectory for a new area of research. Thank you very much.
Thank you, sir, for your thought-provoking inaugural address. Thank you, sir. We will take this in forward.
Thank you.
Thank you. Now I request Dr. Siddhi Jalmi, the organizing secretary of this webinar, to propose the vote of thanks.
Thank you, sir. Am I audible?
Yes, you can.
Yes. Good morning to all the distinguished guests, delegates, and the students. I, Dr. Siddhi Jalmi, on behalf of faculties of the Department of Botany, express my sincere gratitude to our Honorable VC, Professor H.B. Menon, for making his presence in this webinar and giving inaugural address. Sir, your presence has enlightened this program. I'm sure your inspiring words have filled the young minds with all the encouragement and positivity, and they will take up the research in the area that you have mentioned. Thank you, sir, for your immense support and help. I also take the opportunity to thank Professor Vijaya Kerkar, Dean of Faculty of Life Science and Environment, to give her introductory remarks and introducing this webinar in very enthusiastic way. I also extend my gratitude to Professor S. Krishnan, Head of the Department of Botany, Goa University, for cordially welcoming all the distinguished guests and delegates. Thank you, sir, for providing immense support and help in conducting this webinar. This webinar would have not been conducted without the continuous support of all the faculty members of the Department of Botany. So I thank all of them from bottom of my heart, and also the research scholars for their participation and all the participants that are participating in this webinar. Last but not the least, I thank all the speakers for giving their valuable time in this webinar. I'm sure your talk will definitely benefit and encourage all the participants in their respective areas. Thank you all.
Thank you. Thank you, Professor Krishnan. I would leave now. Unmute.
Thank you, sir.
Yeah, thank you, sir. Thank you very much. Thank you very much, sir, for your presence. Thank you.
So now we'll start with the first session.
Yeah, you can
Starting
organize it with the time, and but only thing, make sure that it is recorded.
Yes, sir. That is
It is also inaugural session is also recorded, no?
Yes, sir.
Okay, okay. Make sure that. Then we will start as per the time. No problem.
Yes, sir.
Thank
The
you.
first session is starting at 10:30. So
yeah,
we will meet at 10:30. Thank you.
Okay, okay. Thank you, sir. Thank you, Professor Bhatt. And
Thank you, Professor Bhatt. Thank you. And it was nice seeing you.
Thank you, everyone.
Thank you very much. Professor Vijay, congratulations.
sir.
I came to know only today that you are the Dean of the faculty.
It's a temporary arrangement, sir.
No, all the best.
Thank you, sir. Thank you.
Nice hearing you.
And thanks to all the resource person who could join on this occasion, and all my colleagues. Yeah, thank you.
Krishnan, thank you very much, and I wish you all the. I wish the program all well. Siddhi, same to you. Okay.
Thank you, sir, and we're happy to see you joining our webinar. Yeah, thank you, sir. Bye.
Thank you.
Professor Sharma, are we starting now?
I leave it to the organizer and.
No, I actually means you are the chairperson for this particular talk.
Yes.
And you can please go ahead and start.
Okay. Yeah, I think time was given 10:30, so we are right there, and we can start now. A very good morning to all. And for this session, we have Ms. Ravina Pai as a reporter. So let's start the first session of this national webinar with a distinguished professor of plant science, Professor Subramaniam Rajgopal. And it is my great pleasure to introduce him, a personal friend and fellow student of plant physiology, and more particularly of molecular stress physiology. Professor Rajgopal did his MSc in biochemistry from Sri Venkateswara University, Tirupati. And though he was also registered for his PhD there, but most of his research work was done at GNU with Professor P. Mohanty. Subsequently, he spent 7 long years to carry out his postdoctoral studies at various international institutes like Biological Research Center, Hungary, and University of Quebec at Travis Riviera, Canada, and Arizona State University, USA. After his return from his postdoctoral studies, he joined at University of Hyderabad, where he is working on bioenergetics of chloroplast, protein biochemistry, and protein ligand interaction. And he has published on these topics almost 100 papers in well-reputed journals. And his work is very well recognized in the form of several honors to him. And if you are elected fellow of National Academy of Sciences, Allahabad in 2016, elected fellow of Telangana Academy of Science in 2016 again, and elected fellow of Andhra Pradesh Academy of Science in 2014. And he has been awarded Chancellor Award in 2014 from University of Hyderabad. And he is director and principal investigator of BioNest program of the university. He has visited on various scientific assignment to various foreign laboratories, and like USA, Canada, Australia, Japan, Israel, Russia, Greece, China, etc. So he is very well recognized in his academics. He has received financial grant from various national funding agencies such as DST, SERB, DBT, CSIR, and Institute of Eminence and Potential for Excellence. This kind of grant he has received from national funding agencies. And to his credit, he also has international program, through UGC Israel Science Foundation, one, and another one is GST, GSPS with Japan. So he is doing a great work there. Professor Rajgopal has also contributed to academics as associate editor of scientific reports, guest editor of photosynthesis research, again academic editor for PLOS One journal, editor for Photo Nano Bioscience, and editorial board member for Journal of Photochemistry and Photobiology, a very reputed journal in the field of photobiology. With these words, I invite Professor Subramaniam Rajgopal to deliver his presentation on response of photosynthetic apparatus toward abiotic stress and acclimation process. Professor Rajgopal.
Yeah, thank you very much. Thank you, Professor Prabhat Kumar Sharma, for my long association with him, maybe in 1998. Thank you so much for introducing. And this occasion, I would like to congratulate the Department of Botany for conducting such a nice webinar on advances in plant sciences. And I would like to thank the department head, Professor Krishnan, and the dean, and Dr. Siddhi Jalami and other faculty members. Thank you so much for inviting me for this webinar. I'm delighted to present our work on response of photosynthesis apparatus towards abiotic stress and acclimation process and application. Is it audible, and can you see the slides?
Yes, sir. Yes, sir. It's clear.
Okay. So I will drive through our research for another 40 minutes, and later you can ask me any clarifications or doubts. And as rightly said by the Vice Chancellor, that the recent scenario, we have a lot of global issues, climate issues, and affects the photosynthesis, and obviously leads to change in a lot of photosynthesis process, and yield of the plant productivity reduces. But through abiotic stress, there are advantages as well as many are disadvantages. With that we will discuss about a little bit later. So if you look at the earth formation, you can see here in the left panel, it's almost like reddish blocks as like a bricks like that. There was no living things on the earth. Slowly, if you're going to move on to the down panel of left, and there is you can see here the earth formation, and there will be fire and other things. Then if you go into right panel, right side here, then the globe is formed with the water, and other the living things has formed, and where you could see here right now a green process where we are all happy with the fresh oxygen and other the products that we will see here. Through we'll just discuss about a few minutes for the interest of students the applications of photosynthesis. Probably all of you know, but I just want to give you some overview about the applications. When you see that all these process, it's all obtained from directly or indirectly through the photosynthesis process. Food as well as the whatever food is a vegetarian or non-vegetarian, or most of the medicines, the plant-derived medicines, they're all one way or other connected to the photosynthesis. And the nowadays, because of the natural resources are reducing, so now people are working a lot on to get the renewable energy through photosynthesis, like hydrogen and all the other the ethanol and many other inorganic solvents. And all because of photosynthesis, you get plant productivity, all plant products, and you get a fresh air with a good fresh air is nothing but oxygen. So what are all the other applications apart from this? Of course, the photosynthesis, you can see that with these global warming or whatever, you can see the India Gate here, where the pollution or whatever it is affect the plant productivity. And further, the pigments of photosynthesis are widely used for several applications. You start with food additives, food, all the pigments are chlorophylls, or chlorophyll A, B, or the cyanobacterial phycobilisomes and bacterial pigments. All these one under the keratinite pigments are basically there are several keratinite pigment derived derivative of the keratinite pigments like lutein, zeaxanthin, xanthine, like such kind of pigments gives a lot of the importance for the human needs, human health. Okay, now food industry can use all these the products, food products, and pharmaceutical, how this pharmaceutically how they import, pharmaceutically how they are important, There are anti-inflammatory, neuroprotective, hepatoprotective, hypocholesterolic error, these phycobilisomes are used for hypocholestemic, to reduce the cholesterol. And there are also, recently people are working on, we all know that, the algal system, that is a hematococcus. You can extract the astaxanthin, and the astaxanthin is used highly anti-inflammatory or antioxidants. So likewise, the pigments of the photosynthesis process, obtained pigments can be used for several applications. And the cosmetic industry now, as I said, there are a lot of pigments. As I said, the keratinite derived pigments, especially keratinite derived pigments and phycobilisomes are all used, lipstick, sun protecting creams, and eye shadow palettes. So likewise, and also I said therapeutic, anti-aging, and Alzheimer's, anti-cancer. They're all of them, and the antioxidants, and a lot of the pigments. They're all used as a fluorescent probes. Similarly, in bacterial chlorophylls, also you have a similar kind of applications. The bacterial pigments are also used. You can see that, similar to the previous, all pigments can be also used. More than that, as I said, the natural resources are reducing. Using plants or an algal system, one can develop the using CO2 and artificial photosynthesis. Artificial or a natural light, or also there are other way. I will tell you a little bit later. You can increase the biomass. Through the biomass, you can get oils, that is biodiesel, residual biomass, bio-oil can be used, biohydrogen can be used, bioethanol can be used, and also biogases will be used. And more than that, they can also, all of them in the waste, whatever, the residues can be used as a biofertilizers. And also a lot of, the plant products or algal products can also be used for feedstocks for several other, for agriculture purpose, as well as the feedstocks like a shrimp industry, fish industry also can be used, especially the algal products and in cyanobacteria. But ultimately, you need to have a biomass. Biomass is again through the photosynthesis process. And also there are artificial photosynthesis is emerging nowadays to improve the several other biomaterials, like chips for retina chips, or they're also used as a leaf model. You can artificially you can synthesize the leaf model where you can get all mimicking to the photosynthesis, where you can get all the hydrogen, biohydrogen, and other products can be synthesized from artificial photosynthesis. Okay, now the plants. This is all the review from the literature. There are several, you all know that abiotic stress. Abiotic stress, or temperature. You can see the salinity, heat, drought, chilling, height, light, heavy metals, floods, anaerobious through, and also several other factors. But each one have it cannot be any stress. Biotic stress or abiotic stress is the effect on photosynthesis may not be same. It can vary the the influence on photosynthesis. Let's say salinity, for example. I don't want to give you the entire, I don't want to discuss entire thing, but you can just overview anyway. That will be there in the slides will be maybe saved. You can use it for students can use browse them and get the information. So let's take the upward remarks are showing the upregulation or increase in the quantity, and the downwards are nothing but a down regulation or decrease in the quantity of specific. And then the salinity, if you see here, osmolate synthesis, there will be a lot, and stress response enzymes, and ion transporters. These plays major role and they increase. And if you look at the decrease in that one, disturbed osmotic balance and ion homeostasis and membrane damage will be there, and nutrient imbalance will be there. If you look at the heat stress has a different effect. You can see the high transpiration because the temperature is high, you get more of water deficiency will be there, and elevated evaporation will be there. With these, you get all these damages to the photosynthesis. And where it is increasing, there will be obviously heat shock proteins and trying to acclimate, and induction of protein repair mechanisms also will be there. Likewise, each stress have it is signature on plants. So plants are such a beautiful system where they really acclimatize to most of the stresses. But however, if you give more and stress, so they also will be affected. So further, if you go into the photosynthesis mechanisms here, the photosynthetic photosystem 2 and if you look at the the schematic representation of thylakoids, you have a photosystem 2, a major membrane bound complex, and you have a cytochrome B6F complex. Then you get a photosystem 1, another membrane bound complex, and ATP synthesis. So what it does here, the linear electron transport through water splitting happens, and the electrons are excited, and through photosystem 2 it will transfer through the PQ pole, then it going to the photosystem 1. Finally, you get the NADPH. Through NADPH, the pyridoxine will be reduced. NADPH will you get, and then the ATP will be the one through the lumen side. The protons which is generated from water splitting process, and it is through the ATP motor and ATP synthase motor complexes you get a ATP. These two, ATP and NADPH, are utilized in Calvin-Benson cycle to get the utilize CO2 and get the carbohydrates carbohydrates, and further all other biomolecules, so-called secondary metabolites, and any other metabolic process will be through the Calvin-Benson cycle only it occurs. And if you go into the structural, three-dimensional structures has been discovered, and very well, this from the from photosystem, from cyanobacteria, this is from higher plants. The structures has been almost derived from plants as well as in cyanobacteria, and nowadays in algal also there. But still, water splitting process is not yet resolved completely. I think people are working hard to resolve the water splitting process with high resolution. So then the photosystem 1 structures here, there are the photosystem 2 is almost kind of more similar to the in plants and the algal system and higher plant, but there are some changes are there in coming to the light arresting complexes. In photosystem 1, the synechocystis have a a monomer, and of course in higher plants it is like also similar. Not similar, but there will be there won't be any light arresting complex in photosystem 1 in cyanobacteria. But in a higher plant, it's like a heart shaped. You have a beautiful light arresting complex. You just follow the cursor here down. And it also, if you say the bacterial system, they have only a mimic into photosystem 1. Each one, it has about 18 subunits are there. And if we are going to the chlamydomonas, they have a little very in their structures. If say that in higher plants you have one layer of light arresting complexes, in algal system they have 2 layers of light arresting complex, but rest of all other subunits are almost similar. And this is because the evolutionary they live in deep to harvest more light energy, they need more of light arresting complexes. So now it's going to some more introduction. Plants, you can see the video here. Plants, affects with the various, seasonally they will be affected with the various, the environmental conditions. You can see here green, then it goes to the, senescence, then it goes to the, fall, then obviously, it affects during this process. It affects to the various stress conditions, but still plants are the beautiful mechanism of, they can adapt to all these environmental stresses. So now this coming to the story of our research here, all plants and algal system or cyanobacteria, they expose to various short-term response and long-term response of abiotic stress. In the short-term responses, there will be mostly the ROS will be generated. You can see the blue one, and formation of delta pH, and PQ pool is getting reduced state, and there will be phototaxis, and there will be xanthophyll mechanism, deepoxidation can occur with the short-term mechanism in the seconds to minutes. It's going to minutes to hours. There will be a lot of, like you can say that the kinase activities plays a major role. And there will be going to some kind of change in antenna size, change in PS1 stoichiometry. If you go into long-term exposure to any abiotic stress, maybe in light or it can be other stress mechanisms. And there will be in long-term, you can see a lot of antioxidant accumulation of antioxidants, and there will be a lot of transcriptional changes can occur during this process. You look at all these one. During this process, there will be a accumulation process where it is called non-photochemical quenching process. Non-photochemical quenching, there will be a QE, QT, QZ, and QA that we'll discuss next slide. Now, what is the non-photochemical quenching here? When the photosystem, especially the photosystem 1, now it is also discovered in photosystem 1 also, it is showing similar kind of activities. When photosystem 1, when there is a zeaxanthin is expressed, means the pigment quantity when zeaxanthin is a keratinite derivative, which is increases, you get a QZ increase, that is NPQ1. And there is a specific proteins kind of PSBS and LHCSR3 in algal system. When it gets expressed, those proteins bind to the LHC2 complexes, photosystem 2 complexes, then you get a QE. These are all adaptation mechanism. And there is another one is called state transition mechanism, where STT7 or STN7 in STT7 in algal system, STN8 is in the plants, and where you get a state transitions, the antenna is adjusted. This is another accumulation process. So finally what happens, if it is too much of the light or any other stress, there will be damage that is called inhibition of photosystems. Mostly in highlight you can see. So this is all adaptation or photoprotective mechanism happens in the plants as well as in the algal system or cyanobacterial system. Okay, now I will explain about our work. We are working majorly on these 4 topics: state transition in Arabidopsis and Chlamydomonas, and thylakoid organization in moderate stress in terms of PQ pool reduction, and acclimatization of photosynthesis process, as well as Chlamydomonas, as well as in other plants, Arabidopsis and Pisum sativum. And we're looking at all omics-based applications, and also see that whether how they can accumulate and how the photosynthesis organisms are organized during this process. And another topic which I'm not going to touch, this protein ligand interactions. So now this is from here I'm going to talk about our research. I just want to give you little bit introduction about state transitions. it happens in the plants as well as in the algal system, in the cyanobacteria also. State transitions, like, as I said, we have a differential absorption of photosystem 2 and photosystem 1, where in the fluctuating light conditions or fluctuating stress conditions, they will be adjusted, the balance the electrons from photosystem 2 to photosystem 1 through, you can see that the photosystem 2 will be excited. You can see when light is harvesting, you get PQ pool is getting reduced, plastoquinone pool, and the kinase will be activated. When the kinase is activated, this is the light harvesting complex, the green like line here, and it get phosphorylated. This phosphorylated, as is kinase are like here, radial PQ pool, Q side, that cytochrome B6F complex, and activate LS2 kinase through these two proteins are three specific genes. And STT7 is in algal system, STN7 is in plant system, all plants. So now you can see what happens when it is getting phosphorylated. Any phosphorylated form is not stable. They have to migrate to other place where it does. The migration is nothing but a balancing electrons from photosystem 2 to photosystem 1. It does the electron transport here. There are people who are telling in the earlier stress condition, there is a spillover mechanism that is a different kind of acclimatation. Here in the fluctuating light, in the daylight, in the natural condition, this happens through LHC2 phosphorylation and balance the electrons like this. And what it does, once it is binding to photosystem 1, and again they cannot be, because it has to balance the electrons. Now photosystem 1 will be excited, you see here, and now this will be again the PQ pool is here reduced, now getting oxidized, and you get a dephosphorylation of this phosphorylated LHC2, and again it goes and binds to the photosystem 1. This is what happening through state transition mechanism in plants or all other organisms. So what we did in our laboratory, we have grown the Arabidopsis plants, and the mutants which we have, we have STN7 mutant, and we have probed with several the lighter, the antibodies here. The LHC1, 2, 3 is a trimeric complex, is a trimer. It is three major protein complexes where we probed with antibodies, and where we have seen here and also we probed with the antiphospho 309 with specifically the phosphorylated proteins can be recognized or identified, and where the LHC2B is the major player for phosphorylate. You can see here, this is when it can exactly the same molecular weight, it is matching. That means LHC2 is the one. Of course, the other proteins also get phosphorylated, but LHC2 is getting the major share in phosphorylation. And what we did, we also did a 2D electrophoresis to identify whether it is really LHC, whether it is really phosphorylated. And you can see in the state 1 transitions, state 1, no, that's what I said, state 1 is nothing but here, PQ pool is oxidized conditions. And when it is in state 2 condition, that means reduced condition, that means we used various light conditions to get this status. And where you can see the additional band here, you can see here 1, 2, 3 bands, and here 1, 2, 3, 4 band. This additional band is the one LHC2. Again, we probed with antibody, with LHC2B, where you could see clearly the LHC2B antibodies migrated and phosphorylated. And you can see all of them. If you can magnify a little bit more, you can see here nicely the LHC B band. And we purified all this, using sucrose density gradient, where the state 2 complexes, you can see here, this is the first band is light arresting complexes, a dark band. The second band is photosystem 2, and third band is photosystem 1 LHC2 bands, LHC1 band. along with LHC2, migrated LHC2 is bound to photosystem 1. And the mutant, you don't see any C, almost same. That's what we see, can see here. A sharp peak is nothing but, there is a LHC, additional LHC2 is bound to photosystem 1. And whereas in the mutant, you can see both are same, almost both are same. We showed from the observed spectroscopy also. Then we run the SDS page where you can see clearly this additional band, that is the LHC2, that is entire a trimeric complex. What we said from this, you can go through my laboratory, just in PubMed, you can get the article from one of the journal, where we have clearly said the LHC2 is a major player in taking out the entire trimeric complex and migrated to photosystem 1. So that's what you can see here, the entire LHC2 complexes, where we can see that in state 1 complexes, there are no LHC2 complexes. This means, the picky pool is oxidized. When you see the state 2 complexes, you can see clearly all LHC2 bands are coming out with this third band. Then third band is the one which is a photosystem 1 LHC1, and to that the LHC2 is bound. So similarly, we showed with elevated temperature also with the mutants, there is no change in the structures. When you see this is the fluorescence emissions spectroscopy, low temperature fluorescence spectroscopy, where you can clearly understand this peak is photosystem 1, 2, and this peak is photosystem 1. The dark peak, when you give a slight moderate temperature, there is an elevation of this peak, is indication that LHC2 is migrated. And where we showed here, you can see here, when you go with the various time point, you can see clearly there is a LHC2 is appeared. That is an indication that there is a phosphorylation of LHC2. Those LHC2, phosphorylated LHC2 is migrated to photosystem 1. Even in temperature, we also showed the same one in salt also. We also showed in the chlamydomonas also similar kind of structures. But there are a bit different in chlamydomonas. It won't take 40 degrees, but it is up to 38 degrees. In the control, all these LHC2 are intact. When you have in the various fluctuating light conditions with a short term, you can see that phosphorylated LHC2 migrating to photosystem 1, you can see here. So this is about the model. But in recovery also, somehow we are seeing recovery also, they are still holding with this one because there is a little bit change in the membrane fluidity. It takes longer time to get back to this stage. So this is about state transition. Then I move on to the next the topic, which is about I told you that accommodation process. The non-photochemical quenching is one of the another photoprotective mechanism where we used chlamydomonas, and we also have a mutant generated in the lab that is a cyclic electron dependent. Here you can see here. Photosystem also, when it is stress conditions, the cyclic electron transport is play a major role in order to get the NADPH, where there are two genes, that is a PGR1, and you see here PGR1 is here you can see there two genes play a major role in cyclic electron transport, that is a PGR1 and this a PGR5. There are two genes. So if one gene is not there, if another gene is there will be still cyclic electron transport will be there, but they'll be behaving differently. We wanted to see how these mutants can play a role in photoprotective mechanism. And these are the growth curves where you can see here, this is the wild type. And when you have all these mutants, they reach at the 4th day or 5th day, they're almost close to with these various light conditions here. 50 means 50 microinstants, 250 microinstant, 500 microinstants, various light conditions. And this is the PGR1 with a background mutant, and this is a PGR5 background wild type, and where you can see wild type in the around lower light conditions, they grow good, but if you give a little bit 250, their growth rate is much better. You can see here, this is like 250 microinstance is 50 microinstance optimal light. When you increase little more light, the biomass increases. That's where it is very interesting for the biotechnological industry, where these 250 microinstance is the one can use it for by increasing biomass, for use that biomass for another applications. And we grow them into a state medium as well as in the this autotrophic medium, where both behave same, and we expose them in the spot test, where the 500 microinstance is more feeling like a stress, and in autotrophic and acetate is the carbon source here. Autotrophic means through photosynthetically they're coming from the atmospheric CO2. Here the 500 microinstance, they're not growing, even PGR5 is not growing in 500. That means they're very sensitive to light. The PGR5 is more sensitive than the PGR1. And we analyzed all the pigments by using HPLC. We extracted the pigments, and you can see here all these chlorophyll A and chlorophyll B in various wild type 500 and PGR1 500 and PGR50 500, where the chlorophyll and chlorophyll B there are slight changes, and chlorophyll B is actually increased in PGR5 in 500, and A by ratio is you can see that there is a decrease in A by ratio. And very interestingly, in highlight condition, you can see this one. These you can just follow the cursor. There are significant increase in carotenoids, and you can see here. However, in PGR5, the carotenoids are not increased. Almost very slight increase, but not much. And this is where very interesting with our story. I'm not going to discuss too deep in subject because the students we may not understand much, but we wanted to dig out these keratinite pigments. These are the derivative violozanthine and xanthine and lutein of the keratinite derivatives, where in a highlight the violozanthine is reduced here and zeaxanthin is increased and lutein is increased. So what does it mean? The violozanthine is the one it converts into the zeaxanthin and lutein. Further, you can see here all other the pigments similar in PGR 500 also following same. But whereas in PGR 5, the zeaxanthin pigments are, you can see nothing. There is not much change, and as well the lutein also not change. So this explains that the PGR5 is more sensitive, and for cyclic electron transport mechanism, the PGR dependent route is very important for acclimatization process, and there then we will see other further studies. We checked, these are all, we are not going to show you the entire photosynthesis mechanism. There is a yield of photosynthesis is reduced in PGR5 and PGR1, and little bit in PGR and a wild type also. But we wanted to see that acclimatization process, that means non-photochemical quenching, where in PAM mission you can measure where in 250 or 500 the light, you can see significant increase in non-photochemical quenching. And similarly, in PGR5, there is no decrease. This is the one. PGR5, they not increase, and mutants also the NPQ is not increased, PGR1 and PGR5. But in wild type only, you can see that. So the non-photochemical quenching is inhibited in PGR1, especially in PGR5. That means the cyclic electron transport is a link to the non-photochemical quenching. This is very, very important. So then further, we also checked other, this maybe don't, non-regulated energies loss is a different story here, but I'm not going to discuss about that. We wanted to check more biochemically what is happening to the these super complexes of thylakoids. This is called blue native gel, where we solubilize in beta DM, beta dodecyl maltoside, where you can see this in wild type and PGR1, and this in 50 50 microinstance. I mean, we have not done PGR5, and where you can see that PGR 500 is, and even 250. These super complex are totally, we can say that no, they are disturbed. And what happens? Those disturbed super complex are associated with the light arresting complex. Here, all these super complexes, they came into trimeric form. So what does mean these? Again, the trimeric form is a one. If I want to go into molecular detail, we have did already. These zeaxanthin plays a major role in conversion of these super complexes, or zeaxanthin is induced in destabilization of super complexes. Further, we tested all these. Where we have all the antibodies in the lab. We also raised antibodies in my lab, some of them, especially the photosystem 1 antibodies. And we have, you can see that D1 is getting, we further increased to even 1,000. This is only wild type. And there is, you can see the red asterisk, here you can see that reduced in these proteins. D1 and CP47, and these are CP47 is the one, this is the core complexes, CP43, 47. D1 is the one, again, major reaction center proteins of photosystem 2. And water oxidation is the one, here is the one, the splitting, water splitting process. There is a slight change in there. And we also checked the trimeric form of LHC2, where you can see that the light, they induce, these proteins are changed. You can see this, the trimeric complex are the protein level, they have been changed, and their monomers are also reduced. Very interestingly, there is a protein, as I said, LHC SR3. LHC means light arresting complex. S means stress. R means related. Stress related protein is expressed in iolite conditions. That is an indication that this is directly connected to the non-photochemical quenching. If in highlight condition, you see that, specifically these proteins are increased. So that is an indication that the zeaxanthin is increased. This zeaxanthin is induced the stress-related proteins in order to acclimatize plants as well as in the the algal system, where this protein plays major role. And we also tested all these light arresting complex of photosystem 1 and photosystem the photosystem 1, now of course photosystem 2 we discussed, where the reaction center of photosystem 1 is damaged here, and the reaction is here, PSAB in the reaction center of photosystem 1. And there is a G also little bit changed, G will be somewhere here, and some of the light arresting complexes, you can see that 3 is here. The inner part of the light arresting complexes are, little bit changed. These are all, again, we'll discuss about little bit later, why they have changed. Then further, what we did, we wanted to go into more molecular level, what is happening to the proteins inside, and as well as, how they're functionally connected. So we have isolated all these super complexes using sucrose density gradient. You see here, 500 wild type, 50, 500, in PGR1, and 500 again, PGR5. So in all these strains, here, these are all, again, these experiments are very tedious, and it takes 3-4 days to get all these experiments, the results. Where you can see that these are photosystem, first one is light arresting complex 2, and second band is photosystem 2, and third band is the photosystem 1 LHC1 complexes. In PGR5, all of them are almost reduced. Even light arresting complex has band 2, the phase PS2 and band 3. And we did all these fractions, the light arresting complex, SDS phase of these proteins. You can see LHC2 subunits. There are not much change in the subunits quantity, but there, in fact, the accumulation of LHC2 itself, less than even 550 microinstance of light, but not much change. But whereas you can see that photosystem 2 as well as the photosystem 1 complex, you can see the cursor, there is a significant change. A lot of proteins are down-regulated in 500 microinstant. 500 microinstant is not too much highlight, but it feel like chlamydomonas, they feel like more highlight. But in wild type, they don't feel like highlight. Right? They're almost, you see that, they're not much changed. And we also see that the absorption spectra of all these complexes. Then further, what we did, all these the super complexes, we analyze using Western blots or the antibodies. So we have did all these the 50 and 500 and 50 and 500 in wild type. These fractions were probed with several antibodies, where we can see that photosystem 1 is completely this is the one photosystem 1. Maybe if you want make it clear, F1 is light arresting complex. This one F2 is PS2, F3 is PS1, LHC1 super complexes, and not much change in wild type. On PGR 500, there are slight change in photosystem 1, and PGR 5 and 500, you can see a clear change from here to here. There is a very clear change in the abundance of the protein. We also test with other individual proteins like LHCSR3, PSPS, PSPA, PSF is a control here, and no change in that one. But whereas if you can see that LHCSR3, not much change, but D1 is significant. There is no change. There is no D1. It won't be. We don't expect. But PSA is significantly changed. That means photosystem 1 is affected. That also we'll discuss how the photosystem 1 is affected. I think I'm very close. I think I have many slides somehow. Okay, all these ones now very interesting. What we found here.
Sir, you have about 5 more minutes.
Yeah, I will. That's why I will try to wind up. I mean, I'm not going too deep into other things. Now, what we did here, the proteins there are proteins are aggregated. Whatever when you see that protein to chloropigment ratio, there are the proteins are getting aggregated, where we have seen the confocal microscopy in PGR 500 microinstance light. There is significant increase in the accumulation of proteins. Aggregated proteins are present in PGR5, and there are also in the other highlight conditions. But this is the one. It says aggregation is lead to the change in the photosynthetic activity and other things. And we also checked all these transcriptional factors, various gene expression of all these light conditions. I'm just showing only in wild type here. There are differential expression of each highlight 500, and this is the 1000 microinstance we did, and you can see the differential expression here. The most of the differential expression genes are present in nuclear, and there are less in mitochondria, of course more in chloroplast. And there are upregulated proteins. There you can see the differential expression here, and downregulation there are differential expression. Most of them are almost close. I mean, upregulation as well as downregulation are almost mimicking the same. We wanted to take out only the specific genes of photosystem 2 or photosystems, where there are the green, you can see here the genes like LHCSR3. I also said, you see that black ones are more of more expressed. See that these are all significantly expressed. The light ones are less. The green one is a lesser expression, and if going to the black and reds, there are more expressed genes. Some of the genes are, you can see that some of the specific stress-related genes are the PSBS. There is the stress-related genes are expressed more, that you can see here in highlight conditions. And we also can see that highlight 1 and highlight 2, and there are some specific genes that are expressed more, And this is also we have proved with the protein data analysis. Then further, we also check the transcription factor, I mean the RT-PCR, where we can see the specific genes which I talked about, the conversion of beta carotene to violoxanthin. These genes are increased. You can see here in the PGR5, it is very, very, very significantly increased. Whereas we can see that the zeaxanthin dependent genes are reduced here in PGR5. Now you can see here, that is an indication that PGR dependent zeaxanthin is reduced. That is inducing the change in the the structure of the chlamydomonas structure. We did all again the microtome structures or electron microscopes structure, where the for PGR5 500 there is accumulation of several the vacuoles or the starch granules, and also a lot of lipids here. These are the dark spots or lipids are accumulated to acclimatize to the highlight stress. And we also checked the lipids, where a lot of significant amount of lipids have been accumulated in highlight conditions PGR5, and that is an accumulation of lipids are because of to get the energy alternative energy to acclimatize to the highlight conditions. We also checked all these factors are influencing by ROS, reactive oxygen, using confocal microscopy, where we check. You can see that significantly increased in highlight conditions, the ROS in PGR5. This ROS is induced further autophagy, where ATG8 is involved in autophagy, where you can see here in PGR5 a significant accumulation of ATG8. That is an indication that ROS induced autophagy. Then autophagy also induced change to get the lipid accumulation, where we can see here there is a significant increase of lipids in PGR5. As I said, the PGR5 accumulation is basically coming from PGR 500. That accumulation is of lipids or the down regulation of the membrane lipids of these are the membrane lipids, MGDG, DGDG, SQDG, other PG. You can see here in PGR5 there is a down regulation of MGDG as well as little the DGDG. These lipids are converting into TAG, which we showed here. And also we checked the fatty acid composition of all these protein, all these TLC, the plates, where the in TAG, that means from TLC plate where we extracted, where you can see the omega fatty acid, that is 18, the unsaturated fatty acid is increased significantly in PGR5, and whereas MDG-DG, the several the fatty acids are decreased. Overall, if you can see the lipid composition, the MGDG, you can see the tag is increased everywhere. Tag, that means triacylglycerol. You can see the blue color tag is increased everywhere, and the MGDG is decreased everywhere, and DGDG also decreased. The DGDG is converting into TAG. That's what we showed. Okay, I'm not going to talk about other story here. I don't want to. This is all other one where I will wind up here only highlight stress. What we did here, the highlight state transitions can, to summarize, state transition can induce from fluctuating light and also from the other stresses like a moderate stress and salt stress, where the phosphorylated LSC2 migrate to photosystem 1. That way the energy is balanced from photosystem 2 to photosystem 1. In the highlight conditions, especially in the chlamydomonas, where you can see that biomass increases when the light is more, especially in the white type, whereas in the PGR5 it is behaving differently. They're photosynthetically they reduce, but also even if it is reduced, it is trying to get acclimatized through the accumulation of lipids, and the lipids are coming from membrane conversion of membrane. Membrane lipids are converting into the triacylglycerols, and most of the light arresting complex are downregulated with the change in the protein content. And there are some specific genes. You can see that PSAB, PSABA, and the PSA1, PS1. Some of the specific proteins are upregulated with because the turnover, the D1 and D2, because this is the, because they are all transcription factors. And transcription factors and protein accumulation are different. They will be, behaving differently. So to acclimatize from the highlight, these proteins are upregulated. Okay, the other, several other we have analyzed. I am not presenting all these one. The transcriptome, oxidative phosphorylation, carboxyl metabolism, carbon fixing, photosynthesis genes are some of the proteins are upregulated. And as I said, the LHCSR3 gene is the one which is acclimatation. In the acclimatation process, it plays a major role through non-photochemical quenching. This gene is expressed, and all this one happens because ROS is the one. It is a bad player where it induces ROS, and ROS it changes the photosynthetic activity and other things. But the good thing is there are a lot of biomass is increased, and a significant amount of triacylglycerol is increased. So in the end, what I want to say that cyclic electron transport is very important for acclimatation of the process. If the cyclic electron transport is absent, there will be a lot of changes there. But however, through mitigate or not to change the cyclic electron transport ratio from PGR1 to PGR5, you can get a lot of the accumulation of triacylglycerols, where that can be used as a alternative biofuels. So just I should not miss the acknowledgement. I would like to acknowledge my collaborators from India, as well as my collaborators from abroad, from US, from Joseph Garab and Peter Lambro from Hungary, and my long-term collaborators from the Ichiro Takahashi from Japan, Nathan Wilson. We have a still project with Israel. It's a big project, and of course Govindji is always a well-wisher for all of us. And the entire work was carried out by my students. These are the students, especially the Sai Kiran and Srilata, as well as Ranai and Nisha. These are the people, and Elsin, they are carried out the entire work. And I have several postdocs and present some of the postdocs. I would like to thank all of them. Thank you one and all for listening, patiently listening. And if you have any queries, I will be happy to answer, I think. Thank you.
Thank you, Professor Rajgopal. Thank you for a very nice and summarizing talk on the aspect of photoinhibition and protection and all that thing. And you very specifically utilizing all those techniques prove the point that state transition is one of the protective process. Another, of course, xanthophyll is there, that you used both mutants in Clammy and prove that point also. I was also surprised that the Japanese group is still continuing harping on that PS1 is very important as far as photoinhibition is concerned. Yeah. Okay. Yeah. So let's open this talk for a few questions. If anybody has question, please go ahead.
Sir, there are a few questions posted in the chat box.
Okay, let me see. I don't know. Wait, just a minute. Okay. Participants, good morning. I'm trying to, astronauts and International Space Station are trying to prepare a photobioreactor from seaweed to obtain air and food. I wish to know how will the space condition affect the photosynthetic process in those seaweeds, and also if sir, could you highlight some mechanism by which seaweed would be able to resist space stress? Space. There are actually, and I told you one of my collaborator or my postdoc mentor from Petra Frome, they also did the space studies. What they did, they wanted to check, because the pressure it varies. In the space studies, the pressure it varies, but I am not sure whether the how best they can increase the the seaweed production in space. I am not sure, but these are all one of the studies, but I don't think they can increase the biomass, because there will be a lot of other pressure, low pressure on plants. But there are studies, they're trying to increase the plant yield in space, but I'm really not sure how these are all ongoing studies. But
yes, sir, we can take another question for another participant.
Yeah.
The questions she is asking is PGR5 better than PGR1? And I think I may add here why.
Okay. Now there is other also question, Ravina Pai, where
Yes.
she asked, PGR5 showed downregulation of protein, but showed increase in aggregated protein. So does this mean light stress is directly related to increase in the aggregation stress? Yes.
Yes. So you can collectively mention both the questions are.
Yeah. And both of them, zeaxanthin is not increasing HPLC data, and not what basis can we say it is sensitive to stress? Okay. So what I say that together these two questions, I wanted to say that there is an increase in ROS, especially in PGR5 500 light intensity. So the ROS is induced, the zeaxanthin is reduced because the ROS is damaging the membrane structure. So now the aggregation forms also, the ROS is one, it induces the aggregation of the proteins. Not all the proteins will be aggregated. I didn't talk about that one. There are specific proteins like a reaction center proteins of photosystem 1, and also there are light arresting complex of photosystem 2. This is a very it's a known phenomena that ROS can induce aggregation of the protein. These aggregated proteins are unfolded proteins. They're all they're not favorable for kind of acclimatization process. But I'm not sure whether their aggregation is in the process of the acclimatization. But if it is a reversible, yes. But we also trying to understand whether these proteins are reversible or not. But that is still we are not able to answer that question, because the cells are when you inoculate the cells, they may behave. It's difficult to extract the specific cell in the solution. But the answer is yes, it can induce the aggregation through the ROS mechanism. Sir, any other question? There are somebody raised hands.
Yeah, is there any other question? Anybody want to?
Dr. Sangeeta, yes.
Yeah, your postdoctoral student.
No, she's not postdoc. Yeah, she's.
Hello, sir.
Yeah.
Thank you for an overwhelming talk, and it's always been a pleasure, sir, to listen to you, especially when it comes to your research work. As you have already talked about acclimatization process in plants to various stresses, I understand there also always exists a recovery thing in plants. So just a question and a query to understand whether a similar recovery system exists in Chlamydomonas as well, and if so, how does it actually recover from this kind of stresses, if at all?
Okay, no, that's what I said. In a plant, that leaf, whether it is recovering or not. In the cell, in the same cell, probing that exactly if that cell is recovering or not, that really it is a very difficult task. But that's what I said. We are trying to understand whether the same cell is getting recover or not. But we need to have really magnificent more microscopes where you can see. But there is a possibility of let's take 250 microinstance or even 500 microinstance. They can certainly I want autophagy is also included. The ATG8 is increased. That means there is autophagy, but autophagy is not leading to the cell death. That is a good sign here. So there is a possibility that all the cells are live, the cell death is less here. So they can be, whatever the process, the aggregated process, when can be reversible. Yes, it can be reversible in an algal system, but if you go into deep, like let's take iron stress, we used iron stress. Iron stress, if you go into the repeated iron stress, there the cell death is increased, and there is a possibility of there is a irreversible aggregation of the one. We have reported that one in a BBA paper recently.
I think we can now close this session.
Yeah.
And all it's left for me to thank you, sir, for an excellent talk. Very good talk for students and teacher, because it summarized the whole field and research in the field. And thank you to all the participants. Thank you very much. Thanks a lot.
Thank you very much.
Thank you, Professor Sharma, for conducting this session. And I'm requesting Dr. Siddhi Jalmi to conduct the next session. Thank you.
Thank you, sir. So I invite our next speaker. So our next speaker is Dr. Krishnan Mohan Rai. First of all, I extend my thanks to Dr. Rai for accepting our request in very short notice and delivering talk at 12 in the night there. So this is the US timing now. So Dr. Rai is a plant genetist leader at Cover Incorporated, a biotech-based oilseed company in US. Dr. Rai has received his PhD in biotechnology while working at CSIR and BRI in Lucknow, where he worked on epigenetic regulation of cotton fiber development. After his PhD, Dr. Rai worked as a postdoctoral scientist at Texas University, Lubbock, Texas. During this time, his research was focused on improving the commercial cotton fiber properties. And then Dr. Rai worked as a visiting scientist in Bayer Crop Science at Lubbock, Texas, where his research focused on the development of technologies for freight introgression in the cotton and the soybean. Then he joined University of Minnesota as a research scientist, and he worked on the development of new genome editing tools to ensure this desired editing outcomes, along with the increased editing efficiency. Currently, Dr. Rai is working on several aspects of oilseed cover crop pennycress, aiming to improve the seed protein content and composition and identifying the tissue-specific regulatory elements. So with this brief introduction, I invite Dr. Rai to present his talk. Over to you, sir.
Thanks, Dr. Jalmi, for a nice introduction. Let me share the screen and then I can. Okay. So, can you see my screen?
Yes, we can see so.
Okay. So, as Dr. Jalmi told, first of all, good afternoon to everybody. Or almost good afternoon. There is a time difference here, so I'm a little confused, but it's almost good afternoon over there. So yeah, as Dr. Jalmi told, currently I'm working in at CoverCress, which is a biotech-based startup company, and they are actively involved in developing domestication of a new crop, development of a new crop. So, and about this talk, I was kind of it was too short time to prepare a good talk, but I tried to prepare something. So please forgive me if there is like some lacuna in the presentation. And also, I focus my talk because I know it's mainly undergrads and postgraduate students are attending the talk. So I try to keep it more towards technical technologically, I try to cover the techniques rather than the very deep results and those kind of slides. And there is another constraint because I work mostly in the industry, so I have a confidentiality agreement with them. So most of the time I cannot present my data in the meetings. So I had to cover them with gene X, gene Y, or something like that. So that's something you guys have to bear with me. So my today's talk is focused on a journey of functional genomics, how we started our functional genomics with those tDNA mutant characterization, and then RNAi development, RNAi technique, and then gradually we moved to the gene editing with the discovery of these talents and CRISPRs. So I started my research journey in 2008 in India, at Lucknow National Botanical Research Institute. I started working in a plant molecular biology and genetic engineering lab, which was known to which was very famous for the plant gene regulation related work. And my PI was Dr. Sameer Sawant, who is alumni of Goa University, if I'm correct. So that's why I wanted to show this slide. So when I joined NBRI, so the plant I was assigned, the crop plant I was assigned was like cotton to work on. Cotton is a tetraploid plant. It has a tetraploid genome, and in 2008 there was almost none of the crop plants were sequenced. So definitely cotton was not sequenced because it was a tetraploid. So tetraploid is something last thing people want to touch when it comes to genome sequencing and assembly. And there wasn't any mutant lines available for any molecular study. So my research work was mainly focused around the epigenetic regulation, for which you must have the genome. So the question was how to move forward. One thing I want to say, if I would be more than happy to answer any question in between of my presentation, if you guys want to. So please feel free to interrupt me, because I want to, this is more like technique-based presentation. So if you have some questions, I would be more than happy to answer at same time when I'm presenting, rather than at the end of the talk. So the question was how to move forward. So at that moment, the conventional method or the standard, most of the people, most of the researchers, they have to go through a very standard pipeline. Like, so whatever crop you're working on, take your gene, whatever gene you are interested on, take that gene, do the homology search in Arabidopsis, identify the homolog, use the TAIR database. The TAIR database is basically the Arabidopsis database. Then I find out your tDNA mutant of the Arabidopsis homolog, characterize that, for like do the detailed phenotyping and functional characterization of that particular mutant, and then go back and take your gene from whatever crop you're working and do some complementation studies in Arabidopsis mutant, so that you can, and then you characterize the mutant. So this will give you like whether your gene, so first of all, it depends on whether your gene was, whether the tDNA mutant was characterized or not, it had phenotype or not. And if it had phenotype, whether your gene of interest, like the gene of interest from your crop, complemented the same function or not. So there were so many questions, and it totally depends on your luck, your like what gene you chose, you have chosen. So once you are done with the gene characterization in Arabidopsis, what's next? So the next thing was you have to characterize your gene in your own plant of interest. So there was only two methods available. Broadly, there was only two methods available at that time. These were the popular ones. You have to prepare a overexpression construct, you have to prepare a downregulation construct, and also you can prepare localization construct, like subcellular localization, or to see whether it's a transcription factor or not, to confirm whether it's transcription factor or not, to see where it's in which organelle is functional, something like that. For overexpression construct, this may sound very basic, but I chose this topic, evolution, like tDNA to gene editing. So I want to go through each and every step of this. So in overexpression construct, you have to express your gene with strong promoters to have some kind of phenotype, and then you have to do functional in both of them. So you have to make a construct with the strong promoter. Then in downregulation, you have to make a RNAi construct. You have to choose a small region of your gene, and then you have to clone in RNAi vectors, and then that will produce double-stranded RNA, and then you might know this pathway. So small RNA-based downregulation. So I also did the same thing, and I tried to characterize my gene in cotton. Some of you might not know the cotton is one of the worst crop to work with. I'm saying it worst because that almost ruined my PhD career. Although I was like working very hard, it's double hard to transform the cotton. So to get a transgenic plant from cotton is almost like winning a Nobel Prize. Sorry, it's that kind of making that kind of because in early career, like in your early career, you are not aware of, or you are not technically that much sound. So it's always very hard. So same thing happened with me, but I was lucky to have like cotton, like not few, but considerable number of transgenic plants with overexpression and RNAi line. Then I expressed my gene of interest with 35S promoter, and then I made like you can see, I made RNAi construct with 35S promoter, but I cloned same fragment of my gene 2 times in reverse direction. I didn't show the direction here, so my apologies. But so once the transcript is there, it will make a double-stranded loop, and then I confirm the transgenic plants with the PCR, and then I check the expected expression, change in expression. And in overexpression plant, I found like some of the plants, some of the lines with the good overexpression up to 3 or a little bit more, 3-fold or a little bit more. And in most of the RNA plants were downregulated. My gene of interest was downregulated in most of the my RNAi plants. So the next step, I did phenotyping, and in these are greenhouse grown plants, and I found that the gene of interest GMJ12, Gossypium hirsutum GMJ12, negatively regulates fiber development. So when I overexpressed the number of balls, so it was also involved in the organ development, and the number of balls were there were less number of balls on the plant in the overexpression line, whereas in the RNAi lines the number of balls increased like substantially. And then there was also this is a SL is a span length. So this is a measure of fiber length. So there was increase in the RNAi. The fiber length was increased in the RNAi, and you can see the ball weight was also increased in the RNAi lines. So, and I tried to show the fiber length phenotypically as well. So here you can also see there is a slight decrease in the overexpression line in the fiber length, and there is a slight increase in the RNAi lines. So this difference might look very small, but see if there is an increase in 5%, there is an increase of 5% in length or yield, that's going to impact a lot when it comes to the hectares or acres and acres of cotton land. So this was quite a significant result I got. And then I also tried to check the function of my genes. So the JMJ12 was a histone demethylases, and it was specific to the histone 3 lysine 4. So its specific role was H3K4 Me3 to Me2 demethylation. And in RNAi lines, you can see there is a decrease in the there is a change in the methylation. Methylation. This was done with the anti-H3K4 antibodies specific to monomethylation, dimethylation, and trimethylation. And the methylation in the RNA lines were increased in the trimethylation. So this indicated that the plants, my RNAi and overexpression plants were not only they were transgenic, for sure they were transgenic because of this result, and they were performing, my constructs were performing at the transcript level. So this is like at genomic level they were present, they were expressing and they were performing very well at the transcript level in qPCR results. And then there was a phenotypic difference there, and they were also working very well at the protein level. So I tried to check at each level to make sure the results I'm getting is because of the overexpression and RNAi. So this is brief about my PhD. Then after that time, after graduating from my PhD, I came to USA for my postdoctoral research. And since I was working on cotton, and I joined the Texas Tech University in Texas, this is a place which has a very nice facility for cotton, where you can do the gene, all the molecular work, functional genomics, and then you can get your cotton plants. And then at this place you can, they have almost set up phenotyping laboratory. Then they have a small mill inside the laboratory. So from cloning to making a genes, they can do all the things at the same place. But at when I reached Texas Tech University, after 6 months of my work, I got chance to move to Bayer Crop Science, which is one of the leader in the industry, plant industry, seed company. So I got chance to work there, and I joined them as a visiting scientist. And they were, when I joined them, they were working on the gene editing. They were already using gene editing techniques to make an improvement in the crops. And that was first time when I was introduced to the gene editing. And since I'm on this slide, I just want to tell, like, I worked in Texas, and then from there I went to University of Minnesota, which is a pioneer in filing patent in USA. So they are like very driven to discover kind of place. From there, I got chance to to work in a St. Louis-based company called Covercrest. St. Louis is a place where all the plant-based companies are there, like starting from Bayer, BASF, and all the seed-based companies are there in St. Louis. There's a reason behind putting this slide. Why I choose this map, because you can see snow predicted through Saturday. Today, we are in USA, we are going through one of the biggest storm of the century, winter storm. And this storm is running across, like you can see, 2,000 miles, right from the Texas and New Mexico to all the way to the New York. And this is the biggest storm of the century. So, and I'm sitting right at the middle of that. So I thought this is an interesting slide to use. So just use that. So what about what genome editing methods I was introduced to? So at moment there are 4 different. You guys must be aware of this. So there are 4 different type of genome editing techniques, which are 2 of them are not that popular. Meganucleases and zinc finger nucleases, they are very ancient and almost nobody uses them from long time, not only now but from long time. But so everything is connected. So this zinc finger nucleases, they use this zinc finger nucleases and did some modification, and they that led to the discovery of talents. Talents were very popular just before the CRISPR-Cas discovery. The only problem with talents are like there are 2 molecules of left and right talents we have to use, because talents work in dimer, as you can see. So, and this is a FOC1 nucleus domain. So the DNA binding domain will bind to their target site, and that will bring both the FOC1 domains together. They will make a dimer, and then they will cut at the target region. The good thing with the talents was it was very specific method, because there is two FOC1 has to come together. So even though one binding site is not specific and it's having like a non-specific binding, it's very unlikely that the second right talent would also go to the same place and bind in the exact place where they will form a dimer and they will cut it. So the off-target means non-specific editing was very less in talents, but the biggest problem was each talent is like when you come to the construct, for each talent you have to make 4 kb of DNA. You have to assemble the 4 kb of DNA. So it's a 8 kb of left and right talent. So you are all stressing your tDNA. You are stressing your plasmid vector. And the downstream, there was like more chances of getting broken tDNA inside the plants. So this was popular, but when in around 2012, when CRISPR-Cas system came, so it was so easy to design a CRISPR-Cas9 construct that almost everybody forgot about the talent. But so that's why I gave this title to my this slide, Ease of Doing Business, because it was so easy to, and it increased with as we go downwards, the ease of doing business was continuously increased, and it came to a point where almost it's a simple cloning you have to do to make a CRISPR-Cas9 construct. But when I joined Bayer, and later in BASF, another crop seed-based company, not seed-based, BASF is a chemical company, but they eventually took over the Bayer, and they took over their plants business. So I became part of BASF that way. But so in Bayer, I was using talents. Due to some licensing issues, they didn't have the Cas9 license. This is a big issue in industry, that you have to have license specific to the technique and specific to the crop. So they didn't have license to work with Cas9, so they had to go with the hardware and talents. And we were successful. We were able to generate successful editing. We got successful editing in. I was working on soybean, and I was working on soybean and cotton, and we got more than 300 lines. I had like very strong confidence and sorry agreement with them. So I cannot present any data, and I don't have any data from that where I worked for 3.5 years actually. So, but I can just say I worked with Talon, and it was very successful, and it had like a very specific editing, no off targets. But after leaving Bayer, I moved to University of Minnesota, and where I joined a lab which was where the talents were discovered actually. So Dr. Daniel Voitas is a pioneer in genome editing, and I joined his lab in University of Minnesota, and so they were the people who discovered talents, and they were like also working with the Cas9, advanced Cas9 techniques to do different kind of work. But so I will show those things in detail, how I use for a particular application. So I just want to give a little brief of Cas9, how we design and edit. So how Cas9 works actually. So as you can see, you have to choose a promoter, and then you have to clone Cas9 gene in front of promoter terminator. So that's a one cassette for Cas9 protein, and then there should be another cassette for the protospacers, or in general term guides, or yeah. So initially people use U6 promoter, which is a polymerase Pol3 promoter, and for small RNAs. And so this protospacer is basically the guide RNA, which is 20 base pair, which will decide the specificity of your Cas9 protein, where it will cut. So this protospacer we have to design and clone in the vector, which is fused to the scaffold. So this is a guide RNA, and this is a scaffold. So this U6 promoter will drive the expression, and it will form a RNA, which that RNA will make a duplex structure, and it will go and bind to the target region, which is upstream of the PAM. PAM is so this guide RNA is also called protospacers, and it has a certain need. These protospacers are always next to the upstream of the PAM site. PAM is a protospacer adjacent motif. So you can only design a guide RNA where you have NGG with the SP-Cas9. Nowadays, we have like so many variants of Cas9 proteins from different species. So where PAM requirement is even very relaxed. This is, but I'm just talking about the very first Cas9, like SP-Cas9. So you have to have a PAM site, and then your guide, this RNA will go and sit next to the anneal with the, it will recognize its target, and then Cas9 protein will come and make a double-stranded break at the 4th nucleotide from PAM site. So this is a PAM, so it will come and cut at 4th nucleotide. It's a universally, they will make first cut at 4th nucleotide, and then depending on the repair mechanism, you can have a small deletion, big deletion, insertion, anything is possible. So that's a part of editing outcome, but you can see here PAM and then 4th nucleotide. So then what's next once it made the cut? So how to check that? So mostly what happens is, it's not like you just design a guide RNA and you clone it and you are sure, oh, it will make a cut. It's not always like that. So it's always associated with the word efficiency. So some guide RNAs, they won't they are not very specific. They have like very low efficiency, so you won't be able to see anything in your data. So what we do, we design like multiple guide RNAs for any particular target, and then we test them, we clone them or test them in protoplast. So this is a transformed protoplast from soybean. Sorry, it's a tobacco, transformed protoplast from tobacco in bright field. And so in my construct, I had marker gene DS red, which will give red fluorescence. So whatever you can see in a red color is a transformed protoplast, and this shows very high transformation efficiency. So this is a standard pipeline, like you design a protospacer or guide RNA, then make a construct, then we have to test the protospacer efficiency in protoplast, or you can choose any other available method, but this is a most commonly used method. Once the protoplast is transformed, we have to wait for 2 days, and then we extract genomic DNA from this transformed protoplast and do a PCR with the gene of interest. So if this is my guide RNA or target region, then I design a like 200 base pair from upstream and downstream. We design a primer and do a PCR to amplify this region. And then there are so many methods of choice. You can use different enzymatic restriction enzyme based method, or T7 endonuclease assay, or you can do some high HRM assay. But what we used to do, because nowadays the NGS is so cheap, next gen sequencing, we always try to amplify this amplicon and pull couple of amplicons together and then sequence it. So after sequencing, we'll get like a lot of data, lot of reads, and those reads will be assigned on the tools. That's why I mentioned the analysis, some computational tools, and they will align our result. So you can see the first line is wild type. I have shown the yellow is the target region, and again you can see the edits, and there are so many different type of edits. This number represents the type of edits, not the depth. So there's but there were like from 1 base pair of deletion to almost like if I can say like 20 or 30 base pair of deletion. But I have seen like more than 100 base pair of deletion, depending on what type of construct or what type of a Cas9 you have chosen. So this is a standard result. Then what next? So there are nowadays there are advancement in the CRISPR-Cas9 technology, and so there are so many Cas9 variants. You have to choose based on what your application is. So if you want to just make a standard deletion in your gene, like if you just want to knock out your gene, you just go with the basic Cas9, and it will create an indel. It can create an insertion, or it can create a deletion, and you will get your knockouts. But if you want to do a targeted editing, then you have to do so then there is another method I must say about this. So this is a part of repair mechanism. The editing outcome always depends on what kind of repair mechanism is invoked inside the cell. So it's a chance, but the non-homologous in-joining is a preferred method where your ints will be randomly repaired, and that's like more than 95% of double-stranded breaks will be repaired through this mechanism. Only 5%, that too when you are lucky, will be repaired through the donor. So in homology directed repair, so this homology directed repair has it has an advantage, but you have to provide a donor DNA, and that donor DNA you can edit you can create a few mutations inside, and that will go and do a homology based repairing, and that mutation will be incorporated in your gene of interest or your target. But this is called targeted editing, and the efficiency of targeted editing, or HDR, is very low, very low, 5% or even less. So 5% is something I'm telling very good HDR efficiency. Then there are base editors. So lot of agriculture research in USA is based on EMS based mutations. EMS is a chemical which creates a mutation in the DNA when treated with EMS. So then those seeds will be grown in the field, and they will choose the best performing line. Instead of doing the cloning, targeting a gene, they'll just go forward genetics, and they will get their plant first, and then they will go and map it again. So what gene is affected, or what is the causal mutation? So sometimes you get your causal mutation, but how to, so let's say you have 10 different genes or 10 different plants with traits, different plants with different traits. How to incorporate them inside the one in one plant or elite line? So you have to do, so the common or popular method is introgression, but you have to cross it, and you have to sequentially cross it in combinations, and eventually those will be, or say 5 traits. So you have to integrate them together. That's a time consuming process. So another way is you can use a base editor, or you can use a HDR method, or you can create a base editor. And so base editor will just replicate same, it will create same mutation what we have seen in the EMS based mutation. And so this is a A to T mutation, but the popular mutation is C to T. And there are cytosine base editors, and there are adenosine base editors which can change C to T or A to T. These are obviously nucleotides I'm talking about. So these base editors, again, the efficiency is very less, but they can create exactly what mutation so you want in the plant. And this is again a targeted editing. There are a few more: precision editing, RNA based editing. So, but I have not included those. Then apart from editing, there are some other methods, advancement, where you do the targeted gene regulation. Like, so there is a, one thing I forgot to mention. So the Cas9, so the Cas9 here is an active Cas9. It will make a cut and then it will repair it. But this Cas9 is a dead Cas9. You can see Cas9. So it's a dead Cas9 fused with a deaminases enzyme. So that's how it will create a, so this will just, so scientists have just used a Cas9 complex to target the deaminase to the locus, and then this Cas9 won't do anything. It will just, it's a carrier. Same thing here. They have used dead Cas9, and they fuse a repressor protein. So that repressor protein will go to the promoter region and bind with the, and occupy the, the transcription initiation site, and there won't be any expression. So that's called CRISPR-I, CRISPR repression or CRISPR-I. Same way we have a CRISPR activation or CRISPR-A, where your Cas9, dead Cas9 is, or non-functional Cas9 is fused with the activator molecule. So that will, again, you have to target a promoter region, and your activator molecule will go and interact with the pre-initiation complex. That's a transcription initiation complex, and it will activate the expression of your gene. So, and again, there is another variant which is fused with the epigenetic modifiers like acetylases, demethylases, or anything like that, and that will bring the change in the chromatin conformation. It can based on what you have fused and what enzyme, what is the role of that enzyme that will create an open, that will change the chromatin state. It can bring activation, or it can bring repression based on your experimental plan. So this is a target example of targeted gene regulation. So I have told enough about the techniques. Now I want to show how I have used genome editing in my research. Again, this I have not put very in-depth data. I just try to show the broader aspect of that. But so for that, I would take a case study. So currently, I'm working in a CoverCress. CoverCress is a company working on this plant, pennycress, exclusively working on this plant. And this plant is currently a weed plant. So you can see on the roadsides, not very often, but you can find it in the roadsides. Why this plant? So this is a very unique plant. Brassica member is a Brassicaceae member, which is a Arabidopsis family, and very close relative of Arabidopsis. It has a very small genome and it's a diploid. So bringing the changes in this plant is very easy when it's a diploid plant. And another reason why we chose this one is it has a moderate oil and protein content, which is so. And then there is another few more reasons why we chose this. When I say this is a very important plant, you will see its importance in the next slide. So this is a Midwest USA. The Midwest USA is the very fertile part of the USA, and just like Gangetic Plain in India, and it covers like 40 million acres of land. And USA is very peculiar about its crop cultivation methods or some. I'm not agronomist, but so I cannot use very specific word. But so they grow corn for 3 to 4 months, and then they will leave the land empty. So this barren land will be there for 8 to 9 months of the year, and then again next year they will. They will cultivate soybean. This is not just because they don't want to do anything, but because the condition of the Midwest is very rough. It undergoes very cold conditions in the winters. So almost nothing will survive. So they won't be able to harvest anything. So why? So this plant is empty, but actually empty land of like 40 million acre, we cannot afford to leave this big land empty. But they don't cultivate anything, but there is environmental issues. Since the soil doesn't have any vegetation, the nutrients in the soil will start leaching towards the water, what you call it, aquifers, which is the source of drinking water. And then there is a soil erosion, there is a nutrient leaching. And so what they do, they just throw some seeds, like some plants, which can survive the winters. And they will be there, they will take up the nutrients, and they will stop the soil erosion, and they will be sitting there under the snow of 5 feet. And once the snow is gone in March or April, they will just bloom, and they will be pollinator for the bee pollinator, which is very important issue here. So those plants, they are called cover crops. They will just cover the land and they have no other role. Once in April, the farmer will come and they will just spray herbicides to kill those cover crops. So these cover crops are very important. So our company CoverCress thought about this. So pennycress is one of the it's a confusing names. But so pennycress is one of the cover crop, and our company exclusively works on this. This has a very high oil, like almost 25% oil and 20% protein. Our goal is to increase the protein content to 40%. Oil, it has a sufficient oil. The other problems with this is it has a high uric acid, and it has a high cinegrin, glucosinolate. The sarso smell in India. The compound which smells in India is one of the glucosinolate variant. So that Indian sarso oil is not allowed to. We cannot sell that oil in USA for food purposes because of the glucosinolates. So we have to get rid of this. And this pennycress also has a huge amount of glucosinolate. So we have to reduce the glucosinolate, then increase the protein content, and then reduce the uric acid. So those are some targets. So I will show.
Sorry to disturb, sir. We are running short of time, so maybe
yes,
you can wind up.
Yeah, I will try to wind up. And so how we domesticated this pennycress plant. First of all, this pennycress plant is very easy to transform using floral dip. It's the same method how we transform the Arabidopsis. You guys must be aware with the Arabidopsis floral dip. And then, so, yeah. Then using a standard CRISPR-Cas9 method, we had like two different platforms. One is called discovery platform, where we target single genes and try to see what phenotype it will give. So we have some some target genes in mind. So we targeted them. We use standard Cas9 to hit a single gene and create the deletions. And this is an example. So how wild type pennycress looks like when we did editing in one gene, a cell wall related gene. This, and also that gene was like a gene from phenyl propanide pathway. So you can see the drastic change in the color. So this is basically difference between a rye and a sarso, yellow sarso and rye. These are not exactly the rye and sarso, but that was the difference between the rye and sarso in India. This gene. Then so this was using the standard Cas9. There are so many other examples, but I didn't want it to load a lot of data in the slides. Then I used the SDR repair to introduce another. So we can afford to create a knockout for this gene. So we use a standard Cas9. But so there is another problem. Pennycress is very easily shattered. Shattered means the pods will open with the wind. So you will lose all the crop. So there was a gene called IND. So when you create a mutation in IND, it will be shatter-proof. It won't break. But you cannot create a complete shatter-proof plant. Then your pod won't open at all. So we had to create a subtle mutation. So this mutation was known to create a very smooth kind of subtle shatter plant. So then you cannot use a standard Cas9 or standard genome editing method. So for that, we have to use SDR to create a single base mutation, homology dependent. We provided the same mutation as a donor, and then that was incorporated. And that gave to you can see here, so C22 mutation, T was introduced instead of C, and then the plant, this is a wild type plant. Because of wind, it lost all the seeds. But now the reduced pod shatter, there's a very less loss of the yield. So that was very important. And so you can see SDR was very helpful here. Then there is another advancement. By using a polymerase II promoter, Pol2 promoter, we can do the multiplexing. So we had like our discovery platform for single gene hitting. Now we know, okay, what are the key genes we want to put them together, or gene stacking. So we use these are the 5 genes we wanted to put in a plant. We wanted to create an edit in a single plant. So like different early flowering, local. I have already told about this. So we assembled all the 5 guide RNAs together, and with Cas9, we were able to create a super plant which had all the 5 genes deleted in one shot. and that was very fast as well, because it has just a 3.5 month of life cycle. So we were able to create in a year what this kind of knowledge. So, again, so this is another example of gene editing advancement where you can multiplex. You can go with 10 or 15 genes at a time. Next thing was I wanted to create a seed-specific repression in a gene, but the problem was that gene was, you cannot just create a knockout, because if you create a knockout of that gene, you will lose the winter survivability of so the panicrus is of no use if it doesn't have winter survival property. But the gene, we didn't want that gene in the seeds. We wanted that gene in the plant, but not in the seeds. So we had to, so we cannot use a standard gene editing method. So I went for some advanced NGS method, ATAC-seq, where you can find open chromatin region using some TN transposers. So we, because of time constant, I have to go fast. But so what we wanted to see differential. So what we did is like hypocotyl, root, seed, and pod. We compared the open chromatin regions from different tissues, and my goal was to find a seed-specific regulatory region in that particular gene. And luckily we found, so this is a map of that particular gene, and you can see there are these open chromatin regions. The open chromatin regions are accessible to the transcription factor or any regulatory machinery. So I was able to find a pod-specific and a seed and pod-specific open chromatin region at the 5' and 3' end of that particular gene. Again, we targeted that. We believed that if we hit this and this region, we might get a seed-specific expression of that particular gene. So we tried to cut that using different methods, and this work is still ongoing. And I just wanted to show as a technical advancement. And this is how panicrus look in the field, wheat to future crop. So I wanted to compare canola. You guys must be aware of the canola oil, which is one of the most widely consumed oil in USA and Canada. And this is almost 70 or 80 years back. The canola was a wheat like panicrus. It had same properties, but it was a wheat. Then it took almost 70 years to domesticate the canola into a crop. And now this is a most cultivated oilseed species in Northern America. So same thing we did in panicrus, but with the help of these Cas9 technique advancements, with the help of the NGS methods, fast and cost-effective NGS methods, we were able to achieve same thing in 8 to 10 years. When I say it's mostly 8, but for safer side, I wrote 8 to 10 years. So you can see how the technology helped us, and that too, most of the time took into the what you call it discovery phase, where you have to see or hit a gene and then see the results. They hit a gene and then see the results. Once that database was ready, we just achieved this in 2 years, put those genes together in 2 years. So I hope the broader goal of this domestication is not trying to make sure ensure the food security, because in 2015 we won't be able to feed our population with current rate of crop what we are cultivating currently. So we have to think about the future crops, and this was one of the 10 future crops we were working on. So I was working in a initiative. So I just want to acknowledge my workplaces where I have worked. NBRI, obviously in India, where I did PhD, then postdoc places, University of Minnesota, Texas Tech University, then the companies, industry, Bayer Crop Science and Covercress. And I am very thankful to the all the colleagues, all the scientists I have worked with, and my colleagues, and all the funding agencies like obviously CSIR was the first one to fund, then Walton family, the Walmart Foundation, then Clean Water, Future FGI, I Prefer, and then Department of Agriculture. Also, we had funding from USDA, but I didn't show probably. So thank you all for your patience. I hope I finished on time. And then
thank you, sir. Yeah, but we have a few questions. I hope that we can take a couple of questions here. So the students have asked in the chat box, was there any difference in the cotton texture that you discussed?
Yes.
The second question is there any recommended tool which we can use to design the construct for CRISPR? And
okay. So the cotton which I can I go back? Yeah, I can quickly go back and So the cotton was micronair is basically indication of the texture, the fineness, you can call it the fineness. Lower the micronair, higher is the, some mistake. This is not micronair, this is a micronair, there is labeling mistake. So because that's not the unit, like micronair can't be that high. So lower the micronair value, finer the cotton is. So in the control, the ice strain, and there was a strange observation, but we found both the overexpression and the RNAi constructs. Both the constructs gave us better cotton than the control wild type plant. But yeah, there is something going on, but I didn't have, I was almost in the 6th year of my PhD and I didn't have time to further pursue these results. So I had to move on actually. The second question is like it's a very good question. There are so many tools now, right now available, and the good part is they are available for the mammalian system, animal system, and for the plant system. As a plant scientist, I always want to see plants in the database, but very few design tools have integrated the genome. You have to have your genome integrated to check the off targeting. So yeah, there are so many tools available. You just need to type Cas designer or something like that, guide RNA design, and then you will get the list of the tools. I personally prefer Cas designer because it gives so many data where you have to choose between, because once you put your sequence, it will give you like 100 guide RNA, list of 100 guide RNAs. You have to choose based on the knowledge. So it gives like ranking as well. So yeah, that's a.
Yeah, thank you, sir. There are more questions, but then due to the lack of time, I think they might have a conversation with you through the email. So yeah, so I thank you. I extend my sincere thanks to you, sir, for agreeing to deliver your talk at very last moment. And your talk focusing the advanced technique in genome editing must have opened the minds of young researchers to explore this area. Thank you for your time and efforts. Thank you.
Thanks, Dr. Jalmi, and thanks Goa University for providing the opportunity.
Thank you, Dr. Roy. It's a nice talk. Yes, thank you.
Thanks.
Yes, we'll move ahead to the next session.
Yeah, thank you, Dr. Siddhi, for conducting this session. It's wonderful. And we are moving into next session with a very important speaker. Good afternoon to everyone. And I'm Professor Krishnan from Department of Botany, Goa University. And I have the privilege to introduce our well-known speaker, Dr. Alok K. Sinha. He is very popularly known. Dr. Alok all over the country as well as internationally. I'm happy to see you, sir. And he is presently the staff scientist 7 at National Institute of Plant Genome Research, Delhi. And he has received his MSc from Banaras Hindu University in the year 2000, in the year 1991, and PhD in 1997 from BHU, Banaras Hindu University, Varanasi, and National Botanical Research Institute, Lucknow. And he has received a couple of postdoctoral research. One is 1998 to 2000 as Alexander von Humboldt Fellow from University of Regensburg, Germany, and 2001 and 2 from University of Würzburg, Germany. Then after that, he is joined as a staff scientist at NBRI, National Institute of Plant Genome Research, NIPGR. Since 2003, he is at NIPGR, New Delhi. His research interest, understanding signal transduction transduced by mitogen activated protein kinases, MAPK, in plants. And he is a recipient of all 3 Academy of India. He has received Fellow of National Academy of Science, FNAC, Allahabad, India, in the year 2011. And 2019, he has received a Fellow of Indian National Academy of Science, FNA, New Delhi, India. And in very recently, 2020, he has received Fellow of Indian Academy of Science, FNSC, Bangalore, India. So he is a very eminent scientist. And 2016, he is an elected member, Goa Research Conference. And he is also received, or is having in his credit, a large number of awards. To mention 1998 to 2000 and 2006 as well as 2010. So he has received Alexander von Humboldt Fellowship, that is in the 98 to 2000. Then later he is also re-invited in 2006 and 2010. And he's also received the DST DAD Fellowship in 2007 to 2009. And 2004 to 7, DST Young Scientist, and 2011 BC Deb Memorial Award from Indian Science Congress Association, and 2013 National Bioscience Award for Career Development from Department of Biotechnology, Government of India, and 2015-16 Tata Innovation Fellowship of Department of Biotechnology. And very recently, in 2021, J.C. Bose Fellow Award from Science Engineering Board, Department of Science and Technology, Government of India. So he has received major prestigious awards. And publications, he has more than 70 publications in his credit with the different journals of national, international repute. And he has published papers in the plant cell, the plant journal, plant physiology, molecular plants, Journal of Experimental Botany, molecular and plant microbe interaction, scientific reports, FEBS journals, BMC plant biology journals, etc. And all these journals are having very high impact factor. And also, I'm happy to tell you that our Dr. Siddhi Jalmi, assistant professor, who is here at present in the department, he worked with Dr. Alok, and she was a PhD student of him. And we are very happy to have her also here with us. And thank you, sir. And I think with this, I request Dr. Alok Sinha to give his talk on signal transduction in plants and protein-protein interaction. Sir.
Thank you, Professor Krishnan, for kind words, and you have too many words. Thank you very
Thank
much.
you, sir. You deserve it, sir.
My screen visible?
Not yet, sir. Yeah, not yet.
The problem. No, right?
No, sir.
Okay,
Yeah.
sir.
Maybe you can do it again, sir. Possible.
Yeah.
Sorry if I may.
Yeah.
You have to hit the. Okay.
Sir has left.
Yeah, I saw that. Yeah.
Maybe he's trying to rejoin. Yes. Just we'll hold on for a few seconds, then.
Yeah, I'm back.
Yeah, yes, sir. Yeah, yes, I could see you.
But some problem there. Now I think it will be.
Yeah, please try, sir, once again. Then. Yes, it's coming.
It's coming, yeah.
Yeah, yes.
Is it there now?
Yes, sir. Yeah, yes, sir. Please put it, slideshow mode, and then you can start, sir. Yes, sir. Yes, sir. Yeah, clear. Yes.
All right. So once again, thank you, Professor Krishnan, for such nice words. And it is really a proud moment for me to be here. Not only for this achievement, what you listed, but that I happen to be supervisor of one of the organizer of this webinar, Dr. Siddhi Jalmi. It was indeed a pleasure to have Siddhi in the lab and to learn so much from you during the course of your PhD work. All right. So it's also slight setback to give this webinar virtually, which is planned to be in Goa. So we look forward for a chance to come to Goa, and then because of this prevailing pandemic, we have to stay back in your confined room and talk from the front of a screen, missing all this good time we can have at Goa. Anyway, there's a thing right now for the last 2 years, and I hope that it will be soon come over, and we will come back to this pre-mask days and can meet physically. So this is, I come from NIPGR. For those who have not come to this institute, this is a DBT institute which is situated in the south of Delhi, in JNU campus. And we take, I heard from Siddhi that this webinar is mainly for the students who are doing masters or maybe in the bachelor's. So for those who want to pursue PhD in plants and want to come to NIPGR, we take students by our own exams. The eligibility is there that you have to be masters and qualified any of this national exams like CSIR or CSIR JRF or DPT JRF. Even those who are inspired fellows can appear in our interviews and come here for doing PhD. We have a very good state-of-art facilities for doing plant molecular biology and plant genomic research, and we have almost 29 different research PIs working in different aspects of plant biology. So today for my talk, I have kept it very simple, and as you can see from the title, it's a signal transduction in plants, and then protein-protein interaction. So I have a brief outline of my talk here. I'll start with a brief introduction of signal transduction plants, where I talk about signals, receptors, and reversible phosphorylation. There are many different types of post-translational modification in eukaryotes, but I'll confine only to this reversible phosphorylation because I have my lab interest also on this one of these kinases. And then I talk a few of these techniques used to study protein-protein interaction, which will include FRET, BIFC, SPR, and IS2 hybrid assay. I'll come to each one of them one by one during my course of talk. And finally, if time permits and everything's fine, I'll talk a little bit about my research, what we are doing here at NIPGR. My lab is, the main focus of my lab is to understand this signaling cascade, mitogen activated protein kinase cascade, or MAPK cascade. I will talk about two of this biotic interaction. One is a unique interaction between MAP kinase, and also about this MAP kinase and Xanthomonas oryzae, which causes leaf blight disease in rice. The work done by Siddhi in my lab, and I hope the students might be tired listening to two long talks earlier and another talk before the lunch. I'll ask them to please wait till the end to go through some of the exciting work done by your Siddhi ma'am. Okay, so I start with this, that I know that this is a botany department and people are studying about the plants, and we know that plants are sessile and they seldom get such tender care. So they have to face several challenges while throughout their development, growth and development, and they face challenges both from outside, above the ground, below the ground, from pathogens, from environment, and this poor kitchen of the plants, the leaves, they have to make, or source tissue of the plants, they have to make food and translocate it to the different parts of sink tissues, as they're called, like flower, bud, or fruits, or inside the ground roots. And this is what, and they perceive different types of stress stimuli and transduce this down to the nucleus for appropriate cellular readjustment. And this is what plants are perceiving since ages or centuries. And if you list them, they could be many, like you can have start from gravity, from even wind, from different type of gases, from pathogens, both above and below ground. There are many things which can affect plant growth and development. So what plants have done, they have devised many sensory mechanisms to send such signals. There will be a signal first, which will be then perceived by receptors, and then it goes down to the nucleus by different cascades, which could be, many of them are there, and some of them are very popular. I'll talk about them. So what happens when this entire signal transmission pathway is that, first is that you have to perceive the signal, perception of signals. The special proteins called receptors, they perceive the signals. Then once the signals arrive there, you have to transmit the signals or transmission of signal into cell. Then there is the cell signal cascade, which is also known as signal transduction, and then arrival at final destination. So once it goes to destination, which is largely nucleus, and there is a response, for example, gene expressions and certain other things which regulate the synthesis of different proteins, synthesis of different metabolites, which ultimately affect the physiology of the plants for its better adaptation and survival. And this is what happens in the pathways, that you have a signal which binds to the receptor, and through this cascade, it goes down into the nucleus, interacts with the transcription factor, either activate it or inactivate it, depending upon the requirement of the plant, and leads into the gene transcription, and thus the central dogma of biology follows thereafter. Okay, So I'll talk about signals briefly, then about receptors. So signals, what are general points of signals? You can have a single signal that may start several cascades. Signal may be tiny, but response can be big. Many signals received at the same time, more than one may activate or repair the same cascade. And there is always a crosstalk can occur, and not all cells in organs sensitive to the same signal. So signals are very, very specific for the receptors, and it all depends which signal is perceived by which receptors and which cascades gets activated. Now, what makes a good signal? It should be very, very specific, easily able to travel, is really small, especially intracellular signals must diffuse. It should be created very quickly, for example, by enzymes or pre-made, and then released from compartment in cells, and response really needed for long. So must be turned off. For example, enzymes are resequestered, etc. So you cannot have a signal for long because they keep activating different cascades, which are very energy consuming, and this could be harmful for the plant in long run. Now, signals are made when needed. So you have initial signals, which is perceived by this, and you have a new signal being released. Sometimes signals are released outside the cells also, and then messages being transduced. Now there is always an amplification of signals. So extracellular signals, small, transient, and there is always a second messenger like calcium, nitric oxide, which transmit the signal to other part of the cells. Second messenger are often not produced by receptors, but by G proteins acting on another enzyme. You may have one receptor that activate many G proteins, leading to amplifications, and second messengers activate other proteins, often kinases, that then cause phosphorylation of an enzyme activation at every step. You can have an amplification of this signaling. So if you talk about amplification, this is just a cartoon to show that there's a ligand or signal being perceived by receptor, and then it goes to the second messengers, and you have a kinase which is being amplified. And this kinase can activate many different other proteins, and you have a active signals or enzymes, transcription signals, which gets multiplied in different organs of the cell, of the different organs of the cells. So these cascades are more properly actually wave. And this is another way that amplification can occur. So you can see that you have a cascade here, A, B, and C. You have a B which is activated here, and this component B of this cascade can interact with the component H of another cascade. So there will be a lot of crosstalk, and it is humanly impossible even today to understand everything what is happening inside a cell at one given point of time. With the amount of tools what we have and the advancements of biology, I still think that we need to discover more techniques and to understand this dynamic changes taking place inside the cell at one point of time, or real-time understanding of this dynamisms. We can do this transcriptome, we can do a lot of omic research, metabolome, and whatever you call it. But these are all a time point. You harvest the sample at a time point, and you get an idea of this time point. And looking at different time point, we actually interfere what is happening. But this dynamism of interactions taking place inside the cell, involving lots of post-transfer modifications, are impossible to understand in one go. Now, if we talk about this signal transduction current view, what we see right now, or what we can at least conceive or feel is like that. Something such complex that there are crosstalks almost everywhere. There's a web-like structure going inside the cell, and we can only understand something in one window. We open a window, we understand what is happening here, but at the same time, what is happening here is getting missed out. We don't have, like as I told, the tools to understand all these post-transfer modifications which are taking place in the cell because the receptor has perceived a particular signal. Anyway, this is a topic which will keep on going. Now coming to this receptors, you have a signal first, then perceived by receptors. This is very basic for students now again. This receptor must be located at the cell surface so that they can see the signals. Must be very specific again for the particular signals. Must bind signaling molecule very tightly so that can detect signaling molecules at low concentrations, and must convey the fact that signaling molecule has arrived into the cell interior. There are classes of receptors like G protein linked, ion channel linked, containing intrinsic enzymatic activity, and intracellular. Now, like if you talk about ion channel linked receptor, you have a ligand which binds to this, which is ion channel is closed, or a ligand or signal which binds to this receptor. They open it, and this whatever molecule which is passed can pass through this inside the cell. Similarly, G protein, which is linked inside the with a receptor, once the ligand is bound, the G protein is activated and transduce the signal down to the cells, and how this is how signal is propagated. Now you have enzymatic activity, intrinsic enzyme activity. Once your ligand binds to the receptor, you have an enzyme activity which gives rise to another protein or another, maybe the most commonly used is phosphorylation, and then the signal is transduced. Similarly, if you have, there are lots of intracellular receptors which signaling molecules move through membrane. They are located in cytoplasm or even in nucleus. Sometimes they're also called orphan receptors because they have a sequence similarity with the receptor, but actual function is not known. Can also be activated by phosphorylation always. In plants, some pathogens are apparently recognized by intracellular receptors. So the signal goes inside the plant. There is no receptor at the surface. Once they make new molecules, these are perceived by the receptors present inside the cells, which then transduce the message and ask the plant to rearrange or reappropriate their anticellular activity to adjust to the new environment. All right. So this ligand binding to receptors can be of 4 different types. Just as a classical textbook information. It could be agonist, where it binds to a receptor and activates it. It could be antagonist, where it binds to a receptor, does not activate it, but it stops the agonist binding. So in fact, not activating the receptor. The binding of the receptor to the signal of the receptor is very much similar to that of enzyme substrate activity. Here it is known as KD instead of KM. KD is a measure of the affinity of a receptor for a ligand. So there is a lot of protein chemistry going on with ligand and receptor binding. The receptor sensitivity is also very important. The concentration of a receptor at cell surface is not constant. In general, sensitivity is, of cells to signaling molecules is proportional to the number of receptors. There has to be a desensitization is a common response for receptors, and receptor often exhibit refractory response. So what does it mean? That once a receptor perceives a signal, for the same receptor to perceive the same signal, there is always a time gap. The signal has to leave the receptor, only make receptor available to perceive the next signal. The time gap between the same receptor to perceive the same signal is called refractory response. Yeah. So these receptors, once they have done the job, it has to be recycled. After binding, the receptor has done its job, receptor clustering occurs. Receptor signaling molecules become internalized by endocytosis. Inside of vesicle becomes acidified, ligands are released and may be degraded by lysosomes. Receptors are returned back to plasma membrane, and sometimes vesicle becomes capped with protein clathrin. Sometimes both receptor and signaling molecules are degraded in lysosomes. Depending on the requirement, these are the different functions which takes place inside the plant cell or any cell. Now, we talked about signal, which is perceived by receptor for transducing it down into the nucleus or for starting the signal transition. And this reversal of phosphorylation is one of the most common way of doing this. Now, if you type how many types of post-transfer modifications are there in eukaryotes, you may get an answer maybe in Google, sometimes 200, sometimes 300, and it's very difficult. But we have a lot of information about reversible phosphorylation, and it is also said that it is the most common way of protein post-transfer modification of proteins. But I feel that so because it is easy to study this phosphorylation and dephosphorylation. It's not so comfortable to study other type of phosphorylation molecules like somolysins or nitrosylation, carbonylation. But there are many things which are happening inside the plant. So this is why we have more information about this reversible phosphorylation. So what happens here is there's a kinase which add phosphate group, which is called phosphorylation, and this added phosphate group is then removed by a phosphatase, which is called dephosphorylation. And there are the importance of this. Many aspects of signaling requires changes in protein activity. Activity can be changed by changes in the shape, for example, caused by pH. However, the most common or most well reported where that activity is changed is phosphorylation. This is completely reversible. Kinase phosphorylase, phosphatase dephosphorylate, and two types of kinases, serine, threonine, and tyrosine kinases are known. So serine and threonine are aliphatic amino acids where you have a exposed hydroxyl group which can be exchanged with the phosphate group in the presence of ATP, and same thing with the tyrosine, which is an aromatic amino acids, and it has a OH group here which can be changed with the phosphate. And we have more information about serine threonine kinase compared to tyrosine kinases. Now, that activation of one kinase can phosphorylate many enzymes. The specificity varies. There are the kinase, their action on a particular protein or substrate is very, very specific. Sometimes one kinase can interact with only one protein, or sometimes it's more than one protein. Often, but not controlled by secondary messengers. The structure of this kinase are very, very conserved. Human genome contains more than 2,000 kinases. So there are so many numbers. Now, I just want to focus a little bit on this MAP kinase because I will have some of the results on MAP kinases in the later part of my talk. So this are called as mitogen activated protein kinases or MAPKs, which is a cascade. These are a serine threonine kinase, also known as MAP kinases. Very important because when activated, move to nucleus and activate genes. They can also interact with cytosolic protein and changes their activity. Unusual in that they are themselves activated by a phosphorylation. Action of a specific threonine tyrosine kinase is known as MAP kinase. So this is a MAP kinase, MAPK. There's a upstream, there's a MAP double kinase, MAP mitogen activated protein kinase, which is also called as MAP2 kinase. This MAP kinase phosphorylate and activate MAP kinases, and upstream to this MAP2 kinase, there's a MAP3 kinase, which is called MAP kinase. 3 kinase is more easy. I'll come to this table, this flowchart, which will be more easy to understand. So you have a stimuli or a signal which is perceived by a receptor, and your activates the first component of this cascade, which is MAP3 kinases. This MAP3 kinase phosphorylate and activate MAP2 kinases, and this MAP2 kinase then phosphorylate and activate MAP kinases. Once this MAP kinase is active, they can interact with transcription factor and also with many other proteins in cytosol, and help in gene regulation and also in immune response. Okay, these are the same points which I talked about. And with this brief introduction of this signal transduction, where we talked about signal, talked about receptors, we talked about reversible phosphorylation and particular MAP kinase, I come to this protein-protein interactions. I'm not sure how many of you are working with protein-protein interactions, but I selected a few techniques here, which is being very commonly used in my lab. And if you look at this human genome, we have information about 30,000 genes in human genome, which encodes about 440 to 100,000 messenger RNAs, 4 to 1 lakh transcript messenger RNAs, which then get translated into 100 to 400,000 proteins. And there are more than 1,000 interactions. So you can see how many interactions are there just taking place inside a cell at one point of time. And this is a very, very dynamic process, and they affect a different part of plant or any biological systems functioning. So now to study this, there are several techniques available in the laboratories, but I'll talk about this 4. First is fluorescence resonance energy transfer, or also known as FRET. Second is bimolecular fluorescence complementation assay, BIFC. Third will be surface plasmon resonance, or SPR, and then is-to hybrid assay, which is one of the most common way of studying protein-protein interaction. And based on this, what I'm going to talk about is techniques. I'll show you some experiment which has been done in my lab using these techniques. So starting with this FRET now, seeing is believing is a term which we have heard from childhood, and nature has provided with a certain variant of proteins which can give fluorescence. Like from a jellyfish, Aquaria victoria, we have a Aquaria fluorescent protein, AFP variants, which is very commonly known as, and most of you must be aware of this, GFP or green fluorescent protein, which is being mutated to make it even a better fluorophore donor, which is called enhanced green fluorescent protein. And then you have a variant like YFP, which is yellow fluorescent protein, again an enhanced form of this YFP. You have a cyan fluorescent protein, CFP, enhanced form of that. And also you have a red fluorescent protein, RFP, from Discosoma genus reef coral. Now these proteins, now they have a different property. They have a different excitation and emission spectra. All these proteins are also called as fluorophore. If you look at this table here, you have a CFP which gets excited at 439 nanometer of wavelength, and emission maxima is 476. So this is maxima I'm just talking about. This is your CFP here, excitation, and this is emission. Similarly, all this fluorophore has a different range of excitation and different range of emissions. Interestingly, point to note here is that the emission of one of this, like CFP, may overlap with the excitation of another fluorophore. Now, this property of this fluorophore has been utilized by biologists and devised a technique called FRET, which is also known as Foster resonance energy transfer, because Foster is the person who discovered this. This is the energy transfer between two fluorophores within distance or nanometer scales. It has to be very close. FRET is the radiation-less transfer of excited state energy from an initially excited donor to an acceptor. You have an emission and you have absorption. So next slide will make it more clear. So in 1948, Foster formulated the principle of FRET, hence also named as Foster resonance energy transfer. It is a phenomenon where a fluorescent molecule, the donor, transfer energy by a non-radiative mechanism to a neighboring fluorophore, the acceptor. Now, here you can see that we are using two fluorophores, CFP and YFP. We have a absorption spectra for CFP here, and this is the absorption emission spectra of CFP. If you look closely here, the shaded part, the shaded part is that area which is the absorption spectra of YFP overlapped with the emission of CFP. So you have to have an overlap between the emission spectra of donor with the absorption spectra of receiver. Okay, acceptor protein. So donor must be a fluorescent, and there must be a spectral overlap between donor fluorescence and the acceptor absorbance. Now, what you can do here is that if you want to know the interaction between two proteins, you can do a FRET. How can you do that? You suppose you have two proteins A and B, and you want to say whether they are interacting or not using FRET. And this can be done inside a confocal microscope. So you take protein A and tag it in frame with CFP. Now you take protein B and tag it with, again, should be in frame. There is a fine cloning here with YFP. Two fluorophores, A hooked with CFP, B hooked with YFP. And then co-translate them inside any tissue, maybe onion peel, or maybe in a tobacco or Arabidopsis leaf, which is easy to make a transient transformation. And then you go and see under a confocal microscope by exciting the CFP fluorophore, giving this absorption spectra. In that experiment, we are not giving any absorption spectra for YFP. So if these two proteins are interacting and coming close to this, the emission of CFP will be taken up by YFP, and you can see a fluorescence of YFP. So by giving a absorption wavelength of CFP hooked to protein A, if we see a fluorescence emission from YFP, we can say that protein A and B are interacting inside the organism in plant. All right. So any more confusion can come back to me, write me email, or can call me, taking number from Siddhi. Now from FRET, we come to BIFC, and this is a bimolecular fluorescence complementation assay. This is origin from a classical studies of intragenic complementation of the Laxi locus of E. coli, demonstrating that fragment of this beta-lactam protein that have no enzyme activity can come together, can associate spontaneously to generate an active complex. Similarly, this GFP prime fragment fused to peptide sequences are capable of producing an antiparallel coil that can impart fluorescence. That is, you truncate this GFP, make it into two parts, hook this truncate part in two different proteins, and if these two proteins interacting, the truncate part will come together and give you a fluorescence. Okay. So in FRET, we use two different fluorophores. In GFP, we are using only, in BIFC, we are using only one fluorophore. Now, for a more detail, this is a GFP protein. For example, if you can just have pictorial demonstrations, which is giving you a green fluorescence, complete full-length protein. Now you truncate it into two parts. So it is a 238 amino acid protein. We truncate into two parts, one first from 1 to 154, and then 155 to 238. So this is N-terminal, GFP-N, and GFP-C, C-terminal. Now once we truncate it, the property of giving this green fluorescence is lost. Now, this two truncated part can be used and can be hooked with, say again, you want to check whether there's interaction between protein A and protein B or not. So you clone protein A with N-terminal of GFP and protein B with C-terminal of GFP. And these two will go into a binary vector and then can be, you can transiently transform any epidermal peel or you can go to any tobacco plants or tobacco leaves or Arabidopsis leaves, transform them transiently, allow them to express this for maybe another 24 or 48 hours and check under the confocal microscope. So if A and B are interacting, they will come together and you can see the reconstitution of GFP and this fluorescence. So if these two are not interacting, you will not see the fluorescence. If these two interact, come together, the truncated part or N and C terminal will come closer, interact, reconstitute, and can give you fluorescence. This is how BIFC works. Again, it requires a lot of fine cloning, and then you have to prove that these proteins are actually expressed inside the leaf or whatever you are trying to transiently transform by either doing transform, doing Western with some tag protein, or there are different techniques which can be used. Now, another cartoon just to show the GFP, what happens in BIFC is that you have an N-terminal of GFP and C-terminal of GFP linked with the two protein X and Y and then transform together. If they are distantly apart, they will not interact and there will be no fluorescence. But if they come close to there's an interaction between X and Y, this two truncated part will come together, reconstitute, and can give you fluorescence. This is how BIFC is working. Okay, so we use this fluorophores naturally available in the nature for doing the split and BIFC, and this is so far considered as the best evidence of an in vivo interaction of two proteins. Now again, as I said, there are a lot of limitations and we can do interactions, study interaction of two proteins here, and we can very easily conceive that at any point of time, there will be a complex of protein interacting together. It's not only two proteins interacting for any function. So it may be 5, 10 or even more. But we don't have a technique to understand, study all this interaction in vivo right now. We have a TriFC now also where we can use 3 proteins, but not more than that. In vitro, we can use it for sure, but not in vivo. So still a lot of scope to develop here and to devise new techniques. Now with this, I come to another technique which is called surface plasmon resonance, slightly expensive technique, which is another in vitro study of protein interactions and requires a very fancy instrument. So SPR or surface plasmon resonance is commonly known as SPR. So what happens here is that the resonance which arises when light is reflected under certain conditions from a conducting film at the interface between two media at different refractive index. So it's most commonly is from Thermoc, is an instrument called Biocor. The media are sample and glass of the sensor chip, and the connecting film is a thin layer of gold on the chip surface. Well, let's leave all this what is written here. Let me explain you here. What happens here is that there is an instrument and you have a sensor chip with a gold film. Now you want to study whether your protein of interest has an interacting partner or not. You can use it for identifying something which is unknown, or you can also use it for two known protein interactions, A and B. You know that A interacts with B, you can do many things with this interaction. And this is used not only for studying protein-protein interaction, but also protein-DNA interaction, DNA, RNA, two molecules, bimolecular interactions, and this is non-radioactive study. So what happens here is that you have a chip here with a gold film, and in this gold film you can attach your protein of interest. Okay, so there's a way by which you can attach your protein of interest. This gold film is placed. This chip is placed in a flow cell, which has a prism on top, and there's a light source, which gives you a polarized light. Comes to this prism, fall on this glass chip having gold plate, with your protein and then get reflected back. This gives you a resonance unit. So once you have only your protein, you get a resonance angle, resonance unit, which is a baseline. Now, with this flow channel, you can pass your protein. It could be a protein of, a protein, or it could be a mixture of protein, fruit extract from a leaf extract. And if there's another protein which is interacting with your protein of interest, this reflective light angle will change, giving rise to a resonance signal, which will be recorded. So as it increases, it shows that there's an interaction between two proteins. Now, once this interaction has been taken place, you can use a buffer to wash all those which have not interacted, and can have only those proteins which are interacting with your protein of interest. And this can then be eluted by different salt gradient in a small vial, maybe as low as 0.5 microliter, even 100 microliter, even low, which can be collected in different vials and can be thrown into mass spectrophotometry to understand mass spec to know the identity of this protein. So with this, you can know the identity of protein. And what more, this is another pictorial graphical presence of that you have a glass chip with gold film and your protein of interest, you get a baseline, then your protein interacts here, you have an association, you can get a kinetics of this, and this can be then, sorry for this. They interact, and then this can be dissociated by buffers, and the chip can be regenerated. So the same chip, these chips are slightly expensive, but if you store it properly, can be used repeatedly for maybe a year for different experiments, unless until your baseline goes off. So using this SPR with, you can identify a new protein, interacting partner of your protein. You can find out how specific is the interaction. You can find the concentration, how much of a given molecule is present and active. You can also do the kinetic studies of association and dissociation. You can find the affinity of, if you have more than one interacting protein, which has a more affinity, and you can also identify a binding partner. So almost the entire classical biochemistry can be done with this instrument. So we talked about now FRET, BIFC, SPR, and now I move into his2 hybrid. I have I think I have another 20 minutes or so. Let me see if we can go through it fast. So I have a very nice cartoon here for his2 hybrid. Those who have seen it is fine. Those who have used it, fine. But for the newcomers, it will be very helpful. This is one of the most common way of studying protein-protein interaction or identifying an interacting partner of a protein of your interest. If you want to, if you don't know what is the interacting partner. So suppose we have two proteins, X and Y, and the question is, do this protein bind to each other? So one way to answer this question is to use the his2 hybrid method. And now, to understand this method, we must first consider how gene expression is regulated in yeast. So what happens here is that you have a gene here, you have a sequence, a promoter sequence here, and you have an upstream activator sequence where you have a binding of transcription activator, which consists of a binding domain, DNA binding domain, and activation domain. When this transcription activator comes and binds to this US, this transcription machinery starts and you get your product. This is how it works in yeast. So what is important here? You have a transcription activator which has two components, activation domain and DNA binding domain. These two come together, bind to the US, only then your transcription machinery will start. Okay, and now back to the question. Whether X and Y are interacting. So what we do here is that we make a gene construct in its expression vectors. So we first clone this DNA binding vector. We have a expression vector, one having only DNA binding domain and other having only activation domain. So one protein X is cloned to the binding domain and the Y is cloned with the activation domain. And these two then together will be transfected inside the yeast. And if these two are interacting, your AD domain, which is commonly called as activation domain, AD and BD is binding domain, will come together close to each other and can bind to the UAS, and your reporter transcription will start and you will get the activity of reporter gene. That means these two proteins are interacting. Now, here we knew we had an idea of X and Y protein X and protein Y, whether they're interacting or not. But sometimes, or most of the time, you have a new protein working with that. You don't have any idea which are its interacting partner. Then you have to go. Again, you can use this is2 hybrid as we can also do with SPR. But here, we go for a fishing, commonly called in lab that we are going for fishing. We have a protein X, and does X bind with a protein? In other words, is there a protein Y which is interacting with X? To find out, we are going to go fishing with this two hybrid system, and we will use this X as a bait and try to find out catch this predator or fish Y. We know that this is how this AD and B domain comes together, and your transcription machinery starts. Here we have a LEXZ as a reporter gene which encodes for beta-galactosidase, which this enzyme in presence of its substrate XCal gives a blue color. So this is our reporter gene system to indicate whether there's interaction or not. Now, what we do here is that we take this bait protein and make a cDNA of this and clone it into DNA binding domain. This is ready, and then we transfect into this system. So we have population of a transformed yeast having this X protein with the DNA binding domain in yeast. Now, to find its interaction, what we do is that we don't know the predator, so we take an entire tissue, take out total RNA, cDNA. So we have an entire library of cDNA with this, which is then cloned into activation domain. So entire cDNA library is cloned to the activation domain of yeast, which is then transfected into another yeast population. So we have two populations of yeast, one transformed with protein X in the binding domain, and other with the library of all other total cDNA of this predator tissue, whatever tissue in the activation domain. Now we take out this plasmids and retransform the one which is having binding domain. So we have taken the plasmid out from this activation domain of the yeast and retransformed. So we have now a retransformed yeast which is having both X and Y, probable Y, or AD and BD domain together. Now we take out this, plate it on another plate which is having your reporter gene and XCal system, and if there is any interacting partner, it will be lighted up by the blue color, which indicates that they are interacting, and you can take out this yeast colony, take out this plasmid, get its sequence, and identify the gene or the protein which is interacting with your known protein X. So this is how you can do a fishing. You can identify unknown interacting partner of your protein of interest. So these are a few examples. I talked about this protein interactions. So FRET, BIFC, SPR, and is2 hybrid. I will now go to my research, and the next 10 minutes I'll talk about some of the work and how we have used this protein interactions and signaling in this. Now, as I said that these MAP kinases are a cascade of 3 different kinases linked with the event of phosphorylation, and that gets activated upon perceiving a signal, and they are involved in almost all activities of plant growth and development, right from seedlings to senescence, biotic and abiotic stresses. And in my lab, this is like information about we have about my MAP kinase. They are very specific for the signals which activates them, and these are all 3 kinases, triple kinase, double kinase, and single kinase are multigene family, and they are very strictly regulated by the upstream stimuli and downstream substrate. So one stimuli which activates certain MAP triple K will not activate the same by not related by the other stimuli. So what I mean to say is that these are very, very strictly regulated, and the research what many people working on this MAP are trying to identify this complete cascade right from the stimuli to each component of this cascade, which MAP triple K gets activated, this triple K, which MAP double kinase get activated, and then finally which MAP kinase is activated, and what happens once the MAP kinase are activated. Okay, so this is what we know from the 2 of this model plant, Arabidopsis and rice. We have almost a very high number of MAP triple kinases, low number of MAP double kinases, and slightly more numbers of MAP single kinases. So cascade goes like this. This triple kinase is the controlling switch, where all the signals are from MAP triple kinase gets converged and then passed into specific MAP kinase for its regulations. Now, I just skip this. These are 4 different classes, A, B, and C, which have a typical TEY motif in this MAP kinase activation domain, while D has a group D has a D in between tyrosine and threonine and tyrosine residue. E is glutamic acid and D is aspartic acid. Now, what we do in our here at NIPGA that we are trying to study all these different aspects of macronized activation. We have a submergence stress. We are talking about hormonal stress signaling, abiotic stress, cell cycle, macroRNA biogenesis, biotic stress, and light. But today, I'll confine it to only 2 of the biotic stress. This is one of this unique map-chemical interactions, and this is when I started my lab here at NIPGA, very early days, and started for the first time using this is2 hybrid. So I took one of this map panels, which is called MPK-20-4, because rice has 15 different map kinases. So one of this, which is also known as WJUMK, was very, very funnily named because it is wound and jasmonic acid uninduced map kinase. It was induced by other stimuli, but not by wound and jasmonic acid. And not much information was available for this this map kinase. Let me tell you here, most of the information what we have from group A, B, and C map kinase, mostly from map kinase 3, 6, 4, in Arabidopsis, rice, and also from tomato and tobacco. So I selected this one, which was list studied that time, and started with identifying its interacting partner by doing a is2 hybrid assay. And the work was done by Arshid Hussain Sheikh, that time a PhD student, now is a postdoc in Gulf, Saudi Arabia. And what happened is that we did this interaction, keeping this 20-4 as a bait and using anti-stereal array from rice, and we could see almost more than 500 different positive colonies. So very high number, and we're very happy that yes, a lot of interaction has come up. So we started then identifying each one by taking out the plasmid, making a DNA, going for sequencing and all. Every time when we sequenced, we came out with a Rubisco. So they sequenced on say 20 or 50 of this plasmid, and said again we have got Rubisco. So out of 500, we had almost 470 as Rubisco interacting with map kinase, and then there were some of this another 11 different interacting partners. Rubisco, because by virtue of this high abundance of protein, it interacts with this, and there may be some significance also, but we just looked into the scan with the different interacting partner, and our attention was caught by another map kinase 3, which is going to be very versatile. So far there are no news, no report of a interaction between 2 map kinase. A map kinase interacts with the upstream map double kinase or goes down with the substrate. So we thought that whether this is true or not, and we performed another 1-to-1 is2 hybrid by taking this 3 and 20-4 by swapping vector, AD vector and BD vector, and everywhere we can see this growth of yeast colony in a quadruple dropout media, and also the activity of beta-galactosidase, showing that this MPK3 and 20-4 are actually interacting. We did some more experiments by truncating it and looking at this interaction and specificity, and we could see another is2 hybrid experiment to confirm that yes, they're actually interacting in yeast. Yeast is a different system, no, it's not a plant. So we have to prove that this interaction also takes in planta. For that, we performed first co-IP. Co-IP is another way of studying protein interactions, where you hook these 2 proteins with 2 different, or you can have 2 different antibodies, or you can hook with 2 tags whose antibodies are available commercially. If you can see here, we have a MPK3 tagged with MIC, antibody MIC tag, and 20-4 tag hooked with HA tag. Now, if you do a IP or pull down with one antibody and Western with another antibody, you can see another protein. So like this, we have performed IP with HA here, which is tagged to 20-4. And when we do a immunoblot or Western with MIC antibody, we can see the presence of MPK3, which shows this interaction is taking place in planta also. Now, to confirm it further, we went with FRET. I talked about FRET a little bit earlier, where we hooked our MPK3 with YFP and 20-4 with CFP, it's missing here, and performed the FRET analysis. And you can see that there's a FRET efficiency always. Anything more than 15 is supposed to be a good efficiency, and we found something like 27, which indicated that yes, these two proteins are also interacting in planta by tagging with CFP and YFP. And you have to be very, very careful when you choose your fluorophore partners for studying interaction. And CFP and YFP are the perfect one, most commonly used for fit analysis. Now, further to this, like we identified a unique interaction. We proved that in interaction in yeast, also in planta by co-IP and FRET. But what is the physiological relevance? For that, we went to tobacco, because at that time we were facing problem in getting a rice transgenic. So we went to tobacco and used this tobacco specific pathogen, Pseudomonas syringae tabacci, and infected it. You get a lesion like this in a wild type when you have only pathogen here, or with only your infection medium. Now, when we transform this leaves with either one of this kinase or both together, we can see this lesions going down, or the growth of this CFP or growth of this bacterial count was considerably reduced when you have both this protein kinases expressed at the same time, indicating that this interaction is somehow helping in the combating this pathogen stress. And this was an interesting experiment by Sid, where he has infected the plant and checked it after time. It was found that all these interactions were confined to the guard cell of stomata. So somehow this stomatal way is protecting this infection by regulating this guard cell of stomata. And this was published in BMC. And that's one, signaling of a pathogen activating MAP kinase, interaction between two unique MAP kinase, which was then proved by yeast-2 hybrid, FRET, co-IP, and physiological relevance. Now, next, I just talked next maybe 5 minutes, where we are use this Xanthomonas oryzae, which causes the leaf blight disease in rice, and this is how the plant looked like once it is infected by leaf blight, which causes heavy damage in the rice crop. And we tried infecting this rice plant with Xanthomonas oryzae. This culture we got from Dr. Ramesh Sonti, who was working on CCMB at that time, was very kind to give us and also train us how to infect this plant. And this entire work, which I'm going to talk about, was done by Siddhi when she was in my lab. I think she started in 2010 and worked here for next 5 or 6 years and did a very nice experiments. Let me talk about the experiments now. So what she did is that she infected the rice plant with this Xanthomonas oryzae culture and just checked the qRT-PCR transcript of different MAP kinases, and we found among all this MPK7 also coming up. We have a report of MPK3 and MPK6 playing a role in plant pathogen or abiotic stress, but there was no role of MPK7, which belongs to a group C MAP kinase. So this caught our attention, and also Siddhi was very excited. And we did some more experiments with S2A2 and SA, salicylic acid. But to find the activity of this MAP kinase, MAPK7 in planta, she tagged this MPK7 with a GFP fluorophore, because we have also antibody of GFP available. And still we don't have a specific antibody MPK7. And transform this rice plant very transiently. This was a very elegant experiment where they could manage to transiently transform rice plant with different construct. So transform this rice plants with this MPK7 and then challenge with Xanthomonas oryzae. We could see the activity of a MAP kinase. This is an antibody where you use catches general MAP kinase. Any MAP kinase which is activated will be phosphorylated at threonine and tyrosine residue, PT and PY. And if you use this antibody, it indicates that there's an activated MAP kinase. So we did an IB with this and performed immunoprecipitation or pull down with GFP antibody. So first we have done an IB and then a Western blot, which indicated that this activated protein MAP kinase are indeed MPK7. So this is how she established that this is MPK7 which is getting activated once these rice leaves are challenged with Xanthomonas oryzae. Now this is how it looks like. Well, the lesions formation, we have this wild type rice leaves challenged with this is the arrow which shows site of infection of pathogen, Xanthomonas oryzae, and this is the site of infection of transiently transforming a plant. So this is only with the vector, empty vector. You can see a big lesions here. It has been quantified and shown in the bar graph here. But when we transform it with the OS MPK7, this lesion is abolished or gone down, is reduced like this. And when you silence this with the RNAi, this I think Krishnan talk about this overexpression on RNA lines, which we could do very nicely transiently in rice leaves. And when you silence this MPK7, again, you can see a very strong disease phenotype. So this is how it's established that yes, MPK7 is involved in combating Xanthomatosis oryzae infection in rice. Now, this is another round of experiments just to prove the same thing. Now, to identify the upstream double kinase, she again performed the same experiment of looking at the transcript of MAP double kinases upon challenging with Xanthomatosis oryzae, and out of many kinases, she found this MKK3 getting upregulated very strongly. MKK3, MAP kinase 3. There are, as I told you, there are 8 different MAP double kinases in rice. This MAP double kinase 3 is getting upregulated, and now we used a BIFC to show this protein interaction. So she cloned this MPK7 to the N-terminal of YFP and double kinase with the N-terminal of CFP, and when both of them were co-transfected in the onion epidermal cells, you can see a fluorescence, strong fluorescence here in the nucleus and also in the cytosol. That's been showing, indicating that these two are interacting with each other in planta, so must be involved in the same pathway. Now, to prove that, we have done some IP studies so that they are interacting, and also that MKK3, when included with MPK7 in an in vitro kinase assay, where we look at the radiolabeled ATP phosphorylation, radiolabeled ATP which goes to the substrate. This is the substrate, myelin protein derived from bovine brain. You get a signal when we have this MPK3 and MPK7 together, the activity is more compared to when we have MPK7 alone or only MPK3, indicating that this double kinase is interacting with the MAPCAS double kinase. MPK3 is interacting with MPK7, getting activated upon bacterial infection, and is also activating the activity of MPK7. Now, to prove that they are in the same pathway, what Siddhi next perform is that she transformed this rice leaves with OS MPK3 along with OS MPK7, and also with the RNA fragments of OS MPK7. And as we expected, when we have both this protein co-transformed in this leaf and then challenged with the pathogen, we don't see lesion at all. So that means this MPK3 is upstream of MPK7. Now, fine, you have a stimuli signal, then you have got identified double kinase MPK3, you identified MPK7, the last component of the cascade, but what is that MPK7 is doing? Some experiments. Now, again, I skipped this because there's some mechanism of this here. I'll focus more on this protein-protein interaction. So, she screened many of these worky proteins which are known to act, which are transcription factor, very specific to plant beer. And one of this worky 30 was found to be interacting with MAP kinase 7 in BIFC assay. It's another BIFC assay where you have MPK7 with N-terminal of YFP and this worky with C-terminal of YFP. And when you co-transfect, you can see a yellow signal. I'm sure you can see this in your monitor also. And that MPK7 can phosphorylate worky here. This is the in vitro assay where we express this protein in a bacterial system and do an in vitro kinase assay. And this activity becomes even stronger when we have MPK3 included. The last line, we have MPK3, MPK7, and worky 30. So this MKK3 activates MPK7, which in turn phosphorylate worky 30. And when she express this worky 30 again in presence of MPK7, we can see that the lesions is going down. So she managed to identify from the, we don't know what actual signal coming from the Xanthomatosis oryzae because they have a lot of effector proteins which is being secreted there. We have identified the MAP double kinase, MAP kinase, and the downstream substrate worky 30. What is missing here is MAP triple kinase, which is, I'm looking for some students who can come and complete this work where they can identify which MAP triple kinase is there and which receptors is being perceived by this effector proteins to activate this kinase to give the resistance. And this was Siddhi managed to publish in scientific report. And I thank you, Siddhi, very much for once again for establishing this very elegant technique of transient transformation, which are being used by other students in the lab. Yeah. With this, I'd like to thank all of you for your patience. And before I end, I'd like to thank the funding agencies like DBT, SERB, and my institute, CodeGround, NIPGR, And to the young students, I would like to end with this remarks that there's still a lot to be explored and lots of new techniques to be established. And the biology needs a lot of fresh brain to illustrate with post-translational modification and signal transition pathways. So it's an area which is wide open, lot has to be done. I mean, we have achieved a lot, but still I think we are standing in the bottom of the mountain and we still have to climb a lot. And before this, I end and thank you all. I'd like to thank my students who are working right now in my lab, and we work on rice and also in Arabidopsis. And if anybody wants to have more information about our work, I'm most welcome to contact me. And thank you all for your presence hearing, and I'll be happy to take questions if any. Yeah.
Sir, you're not audible.
Huh?
Krishnan sir.
Oh, I was audible, right?
Yes, sir. You're audible. Krishnan sir is not audible.
Okay, I thought to the screen only.
Can you hear me?
Yes, ma'am.
Thank you very much, sir, for your wonderful talk. It is a very informative, and I'm sure students and all of us have gained knowledge through your comprehensive account of presentation. And we have a few questions to put in the chat box.
Yeah.
So I'll just read out for you. How specific are these receptors and signaling molecules to various environmental stress?
Yeah. Interesting question. I mean, see, there are, what we know still that most of this environmental stress are perceived at the cellular level by cellular permeabilization, by change in pH and all. And actual receptors are not being discovered so far. There are indications of certain proteins acting on the receptor, but those are not only the specific proteins. But if you look at the downstream events, there are different cascades activated by different environmental stimuli. So we can conceive that there are different type of receptors for different stimuli, and they are very, very strictly regulated because they deactivate different downstream cascades. Yeah.
Okay. And another question is there any method to study total or multiple protein interaction occurring in the cell?
This is what I'm trying to instigate, and good that Vinayak, you asked this question. No, it's not there. Yes. We can do only for 2 or 3 proteins, and I'm looking for somebody who can come and tell me I can devise a technique for you where you can do this study of more than a complex of protein interactions in one go in vivo. You know, if you type something like rainbow, something like a rainbow in Google, you'll find some nuclear sensory mechanism going inside the brain, attached with different type of, I think, dye and all, which gives a signal like a rainbow. This has been done in the animal system, but nothing for proteins as such. So I have been talking with many of my bioinformatics colleagues also to devise a protein or this fluorophores, truncate them into 5 or 10 parts, which can come together and reconstitute. If they can give me an idea, we can do this experiment in the lab and prove that. Yeah, we are waiting for that. Some new technique has to come up.
Okay, sir. And one more question. Plant hormones are known to influence signaling through MAP kinase cascade. Any particular method to study this relation under the plant stress condition?
Very sure. Yes, why not? Yeah, we have information about, and I'm working in the lab also, we have a very nice information about oxygen signaling and cross-road oxygen signaling MAP kinase. Different stresses activates different type of hormones, and you know one of the most common hormone is ABA, which comes up in cold and heat stress or even in drought. And there are specific mechanisms that activated by these hormones, which then the activated MAP kinase then goes down and interacts with the transcription factor like ABA responsive factor or oxygen responsive factors and modulate the genes which are those hormone specific. So there are a lot of information available in this, and people are trying to study this in different aspects. We have identified an ABA responsive MAP kinase which interacts with the ABA responsive factor and regulate them during seedling development, during flowering, and also during drought stress in Arabidopsis. Yeah, this can be done.
Okay, sir. Sir, one more question just to put. And can SPR be used to study multiple protein interaction?
No. Again, because you have an SPR where you have chip in the gold film, you have immobilized only one ligand, one protein of your interest. All right. So this one molecule of the protein will only catch another protein. One molecule of another protein. So if you have 10 different molecules of protein attached to this, for example, on the gold film, it may have 10 different proteins attached individually to each molecule of the protein. So not multiple proteins interacting together. Yes, by using SPR, you can find that there are multiple interacting partners of my protein of interest.
Okay. And Professor Sharma is raised hand, and Professor Sharma, you can directly ask him.
Yeah, that would be good to have.
Yes.
Yeah. Okay.
And I just forgot to mention thanks to Professor Janardhanam, who happens to be a very old, long time friend and a mentor to me also. Yeah, Professor Sharma, please.
Professor Sinha, very nice talk. And actually, I just wanted to, get your thought on that. That this, salicylic acid is a phenolic hormone which is involved in this signal transduction with regard to pathogens by producing the burst of ROS. Right, but we also are noticing that some of these hormones are able to provide protection against ROS. So what do you envisage the possible mechanism?
See, you pointed out salicylic acid. Let me tell you the interesting story about how this map panel studies came into being. It was in 1976-77 when plant map panels started getting reported in the literature. And they identified 2 map panels activated. That time they were only doing the activity of map kinase, followed by, because animals are always ahead of plant science, so there was a technique to identify the activity of map kinase. And they find one getting activated by wounding, other by giving salicylic acid. So they called it WIPK and SIPK, not MAP kinase. Wound-induced protein kinase and salicylic acid-induced protein kinase. Okay. Now we know that this salicylic acid activates certain MAP kinases in both ribosome and rice. You have a signal which is perceived, and one of the first cellular activity is production of ROS. This ROS acts as a secondary messenger and activates MAP kinases. They are known. So it is ROS acting as a second messenger after perceiving the signal, activating MAP kinase cascade, which then goes down, interacts with different type of transcription factor, also those involved in the biosynthesis of hormones, including salicylic acids, which in turn again activate or inactivate MAP kinase depending on the recording plant. So this all are interlinked and there are several reports of crosstalks. So ROS, MAP kinase, hormones are very nicely interlinked.
Okay. Thank
Does
you.
this answer your question?
Yeah, to some extent, yeah.
Okay, if there are no questions, and but still you can contact sir anytime through his email, and then you can discuss with him. And once again, sir, this is a very nice talk, and really we enjoyed with your interaction with all of us. And I'm sure we really welcome you to Goa anytime.
Sure, I will be that.
When the things are improving.
Thank you very much.
Yeah, come sir, and we'll be happy to have you here. Yeah, right. Thank you. Yeah.
Okay, once again, thank you all. Thank you to you, Professor Krishnan, and to Siddhi and all the other faculties of Goa Botany Department.
Yeah, thank you, sir. Yeah, thank you.
Thank you, sir.
And just an announcement of our afternoon session. Then, so we will break for lunch. Then we will assemble back at 2:30 for the next session. And I'm sure Professor Vijaya Kerkar will be chairing this session. Next one. Will all of you please assemble back? Maybe by 2:25 you can join back so that we can start at 2:30.
Yes, sir.
Thank you. Thank you. Thank you, everyone.
2:30. So 3.5 minutes more. Welcome, Dr.
Yes,
Vaibhav. Good afternoon to you all, and a warm welcome to this afternoon session. Seaweed farming is the fastest growing aquaculture sector and can benefit farmers, communities, and the environment. And it also accounts for significant percentage of global mariculture. It delivers a broad range of ecosystem services, providing a source of food, natural products across a range of industries. It also offers a versatile nature-based solution for climate change mitigation. So, scaling up seaweed aquaculture will help in increasing bioeconomy. India, with her long coastline and with a rich seaweed biodiversity, would be the asset for the seaweed farming. India is also expanding in seaweed production. Efforts of scientists, fishermen, and encouragement from government is slowly bringing about a blue revolution through sustainable developments. However, there are many associated problems with it. Some of them are area specific, ecological, etc. And to address that, we have a very eminent personality with us, Dr. Vaibhav Mantri, Principal Scientist and Divisional Head from CSIR, Central Salt Marine Research Institute, Bhavnagar, Gujarat. CSMCRI, Gujarat, along with her regional centers, is well known for seaweed farming of Capapicus and many more genera in Gujarat and Tamil Nadu coast. Dr. Vaibhav Mantri, as I mentioned, he is heading the division. He is MSc from Pune University, now known as Savitribai Phule University. He completed his PhD from the same institute, CSMCRI. His main focus is on strain development, artificial seedling production, protocol to reduce epiphytic infestation, etc. He has 2 patents, over 80 research publications. He has 2 patents and more than 80 research publications to his credit. And his 8 students are working with him for PhD. He has earned several laurels and awards. Some of them, the noteworthy are award for S&T Innovations for Rural Development by CSIR, for his work on seaweed cultivation and value addition, and recently he received award from DBT on biotech product process and development and commercialization, on the cultivation of Gracilaria dura in the sea and process and extraction of agaros thereof. So with this introduction, I welcome on my own behalf as well as from the Department of Botany, Goa University. Dr. Vaibhav will be speaking on epiphytism, diseases, and grazing in seaweed aquaculture. Welcome, and I request you to go ahead with your presentation. There will be a question answer session after you finish, and that's all from my side.
Thank you so much, madam, for your kind invitation and introduction also. Am I audible, please?
Yes, very much.
Okay, thank you. So I will I'll straight away go to my presentation.
Yeah.
As madam, still it will take 1 or 2 minutes. As madam said, seaweed cultivation and seaweed farming, all these are very important for social development. And as she rightly said, there are multiple problems. And I'm going to talk about one of the problems, that is epiphytes, diseases, and grazing. So I club all these 3 points into only one problem that basically kills the plant, to whom we basically want to cultivate. I'm going to talk about it. And as I received the invitation, I understand the audience mostly is MSc and PhD students and researchers who are generally to be a botanist or biologist. And I am bit more specialized with this topic. So I thought of keeping everything very simple, and I would be very happy to interact with you more. So I have kept my slides very simple, and at the end I would be happy to take your questions. Whether my slide is visible to all of you?
Yes, yes.
Okay.
Yes, sir, it is visible, sir. Can you make the slide in a full screen mode?
I made it full screen, but I don't know why it is not seen as a full screen.
Okay. Okay.
Whether I have gone to second slide. You are seeing my second slide, which is seaweed diversity, or not?
Not yet, sir.
We can see your first title slide.
This is sometimes problem with now is it first slide or second slide?
Second slide.
Okay, I think I'll better go like this only, even if it is little inconvenient. Otherwise, most of the times they don't move, slides don't move. So as I said, anyway, I'll share my presentation to all of you so that if somebody wants to read, letters would be very small, they can read it. As many of you might have, audience is mostly from Goa. I'm sure all of you might have seen seaweed on many occasions, and you are botany students, so I'm sure some of you might have also done practicals. If you see seaweeds, what are seaweeds? It's very generic term, and seaweeds are not same. Phylogenetically, they are very, very different plants brought together under the umbrella name called seaweeds. And many of the people where I talk about, applications and things like that, at the end they always call and ask me why they are called seaweeds if there are so many applications. Unfortunately, seaweed has a negative connotation to this term, but unfortunately they are worldwide known as seaweeds. I would better say it as marine macroalgae. So this heterogeneous group is basically classified into three categories, and that also is again artificial based on the pigment and food reserve. And these pigments are of various colors, some of you must be knowing, and they are classified as rhodophyta, phaeophyta, and chlorophyta. And if you see a global database, there are more than 10,000 seaweeds reported. And if you see India, the recent checklist was by Botanical Survey of India by Rao and Gupta. It reported 865 species of seaweeds. They also have included some of Cyanophyceae group. I have purposefully kept them aside because Cyanophyceae, they are not, or Cyanophyta, they are not seaweeds. They are basically a specialized group, and that comes under algae but not under marine macroalgae or seaweeds. So as I said, I'm not going to read. You all can see I have given worldwide distribution or records vis-a-vis Indian records. Always, Rhodophyta is a bigger group. Basically, Rhodophyta, these red algae, they tend to grow at deeper areas, and that's how their diversity is always high, because you have deep ocean, lot of deep ocean habitats, and that's how Rhodophyta is always more. It represents about 50% of the existing diversity of the country, followed by Phaeophyta and Chlorophyta. Chlorophyta mostly occurs in the intertidal region and very near to the shore. So this basically about its classification. I thought of showing some photographs for those who might have not seen seaweed or who might have not aware of the diversity of seaweeds. These are the photographs. You can see there are green, very beautiful colored shape, and brown and red algae. Some of them are economically very important. And as I move to my next slide, which I always give in any of my presentation, many of you might be not aware that we do use seaweed products or seaweed component every day. I'm going to talk about global production shortly. And before that, let me tell you that whatever we are harvesting or cultivating today in the world, more than 70% of it goes in food application, in food, not even application, direct food consumption. And most of the Southeast Asian countries, they eat seaweeds, especially Japan, Korea, Vietnam. So these all countries eat seaweeds, and as I said, more than 70% of it goes in the food application. Rest of the 30% production goes into phycocolloid production and application of phycocolloids. But in India, if you see, 100% is about hydrocolloids, and these hydrocolloids have diverse application. You can see from pet food, cosmetic, boot polish, baby food, jam, gels, jellies, etc., etc. Everywhere, basically, seaweed products are used. I very fondly give an example. All the lady members who are present here in the audience, I always give this example. If you basically prepare a curd, it's a fermentation process. When you prepare a curd in your house, next day or next to next day, lot of water comes out. But if you buy curd from market, you don't find such watery curd. That is basically because of addition of small amount of carrageenan to it. So most of the dairy products, carrageenan is used, and they absorb lot of water, so they form a gel, and that's how they have a diverse application. Let us see a production trend. A global seaweed production is estimated by FAO, Food and Agriculture Organization of United Nations, at 32.39 million metric tons weight, and its value in US dollar is about 13.3 billion. Now you probably can correlate the expanse, of seaweed production and its value. Although I said there are 1,000 species reported worldwide, or even 10,000 species reported worldwide and 100 species in India, but there are only 200 species for which some kind of application is known, and about 6 to 7 species or genera are being cultivated today. So you see how small part we could basically domesticate it and also started production on commercial scale. If you see, most of it before 2010 scenario was very different. I'm going to come next slide and show you what kind of seaweed cultivation is happening. Earlier there were brown seaweeds, mostly Undaria, Laminaria. They were dominating the global trade. But when these countries, especially Indonesia and Philippines, very aggressively started farming, and the trend has been reversed, and red algae basically, especially Euchema and Kappa ficus, they came to a forefront, and now today the maximum production is come from Euchema or Kappa ficus, that complex, followed by Laminaria and Gracilaria, etc. You can also see that not many seaweeds are doing farming, and as I said, it's more popular in Southeast Asian countries. So the Southeast Asian countries are the one who dominate. Half of the production today in the world comes from China, followed by Indonesia, Philippines, Korea, Japan, Malaysia, etc. India also, we produce seaweed. As madam has told in the introductory remarks, Tamil Nadu we are cultivating, some parts of Gujarat, Andhra Pradesh it has taken up, but India still stands 13th position today in the world production, very low. And government has come with very big plan called Pradhan Mantri Matsya Sampada Yojana, in which for the first time around 600 crores have been allocated only for seaweed farming and processing, which is very good, as far as our country is concerned. Let us go to commercial cultivation ventures. I have given here some of the photographs just to make you aware how cultivation is happening. The left-hand side first photograph is from Tanzania, and it is of Kappa ficus cultivation. You can see it's called off-bottom farming. It is done at very near to the bottom. There are wooden pegs, used as an anchor, to which ropes are basically tied, and seaweeds are cultivated. Right hand side top photo is about brown algae, as I said, Undaria, that kind of seaweeds. This is from Europe. Europe had just taken up in seaweed farming, and a lot of cultivation is happening, especially in Canada and Ireland, that part. And then there are tank culture. Mostly tank culture is popular in Israel, and they are doing it for two algae, Gracilaria and Alva. Then there are other two photos. A middle photo is of Monostroma, that is another edible algae. The photo is from Korea. And the last one, bottom photo, you can see it's such a huge cultivation farm is there, and that is from China. If you see the global aquaculture as such, about 50% of the global aquaculture is represented by seaweed, and it is expanded to something about 2000 square kilometer area, and it has 6.2% annual growth. So this is what is about cultivation. Now I'm going to the next slide, where I'm just trying to give you the idea that the seaweed farming is now also looked, as I said, if you remember, 70% is going for food, direct food consumption. So it is now looked upon as a future food. We all know that our population is increasing, and we have a limitation, because whatever our food comes is through agriculture. Agriculture land is limited, water resources are limited, and so people need to think about future food. And the seaweeds are now looked as a lucrative option. It is basically a renewable resource, and today we have 7 billion people. In 2050 we will be having around 9.7 billion people, and basically we need large amount of food. So seaweeds are seen as a food option. Why seaweeds? Because they provide livelihood. The techniques employed are all sustainable techniques. It also promotes biodiversity, because we are taking the biodiversity and expanding it. There are no chemical inputs, like any agriculture crop where you have to use herbicide, pesticides, fertilizers. In sea, everything is present and water is plenty. So you don't need even water also for irrigation. And it's an organic production. It gives better food security than any of the land plants. So all these applications, all these characters make it as a very emerging food source. There, some of you, I'm sure, might have heard about United Nations Sustainable Goals. There are 14 sustainable goals, and this slide essentially shows you where seaweeds can fit. I'm not going to talk. You can read yourself. There are so many applications. Let us see in terms of ecosystem services. What is the most, teething problem is carbon. A lot of carbon dioxide and carbon monoxide and greenhouse gases are being emitted because of our industrialization. Seaweed cultivation, as being a photosynthetic organism, you all know that it is capable of fixing carbon. So it is looked very promisingly looked upon as a carbon fixator. Also, there are a lot of polluting industries, or you see that all the big cities are situated very near to the seashore, and all these water, it goes into sea and it brings a lot of nutrients. So they are also seen as nutrient absorbents. As I said, they give food, they are useful in feed, etc., and energy, and so many things. So it basically is an apt example for fulfilling the sustainable development goal also. As I said just now, you can see here, this is a circular bioeconomy. Madam was talking about bioeconomy. As I said, the seaweeds, basically a very recent study at Australia had shown that as small as 1.5% seaweed is added to the feed, cattle feed, then their they basically emit methane. All these rumen containing cattle, if you add this seaweed in their feed, 70% of methane emission is basically stopped. So that is a very good application. As I said, fertilizers, they can come from seaweed. Fuels, people are looking at seaweed sugars to ferment and produce bioethanol, etc. And as I said, carbon sequestration and storage. So all these basically gives a circular bioeconomy perspective to this bioresource. Now coming to my talk, which, as I said, when you have seen so many numbers, these many tons and all, many of you are from agriculture background, and being a botanist, you know every plant has a pest pathogen. So same it's true for seaweeds. So let us see what kind of epiphytes or epibiotic communities thrive on seaweed, and they act as their pest. So there are, starting from viruses to vertebrates, invertebrates, you know, seaweeds itself, they can be pathogenic, not pathogenic, but epiphytic and detrimental fungi. So these groups basically forms biotic communities around seaweed, and they act as a pest. These communities are classified into two forms: macro, as you all know, which is visible to your naked eye, and micro or meso. But when I talk all about this and give examples, there are several kind of groups which are not studied yet, you know, in detail. For example, slime molds. Not many studies, rather I have not came across any of the study which describes slime molds on seaweed, or you know, these kind of things, or protozoans, for example. There are no studies. So these are some of the gap areas one has to study. When you talk about epiphytes, there are 5 types proposed. You can see here the diagram is given. As all of you are botanists, you know, when you take a section of a stem, you have various kind of tissue, starting from merisstoderm to cortex, you know, to medulla. So basically, how far that epiphyte has penetrated, they are classified into 5 types. And type 1 and type 2, you know, it does not result into host tissue damage, because they are mostly at the surface area only, and they don't do great damage. And type 3, you know, it basically cuts into your protective tissue and then damage the cortical cells, while type 4 and 5, they basically disintegrate entire thallus. So considering the complexity of the seaweed thallus, these are the 5 different you know, types of epiphytes. How do they act? Let us see, there are 2 cycles. One is biological and one is chemical. So biological, any biological entity requires some sort of spore to germinate. Same is true with seaweeds. Seaweeds rather has several types of spores, I would say. Some this example I have taken from alga. Those who know life cycle of alga, they know that there are 2 types of reproductive bodies, either gametes or spores, and both of them are capable of germinating and regenerating. In red algae, there are a couple of different types of spores. So in biological form, basically this spore is required, which has to settle and then penetrate into the tissue and grow. You also have to visualize that this all happens inside the water, where water currents are coming and going, and settling of spore and establishing and germinating, it's very difficult in moving water. But then how it must be happening? Most of the time, which are in the intertidal area, water recedes during low tide, and at that time basically they establish, and then they attaches, and these are very fast you know, phenomenon. Takes place in couple of hours, not even more than 1 hour, I would say. Entire process of regeneration and germination takes place. So this happens in biology. And if you see in case of chemicals, chemically, as many of you are aware, all these, as I said, they have polysaccharides, and these particular spores or epiphytes or bacteria, pathogens, they have a capability to disintegrate those polysaccharides, because to penetrate inside, and these polysaccharides are cell wall material, basically they have to dissolve them. So all these agarase, cellulase, alginase, etc. I have not mentioned carrageenase, but carrageenase also is required. So carragenophytes can be penetrated. They basically gets dissolved, and then they penetrate, and then they produce secondary metabolites, and lot of crosstalks, chemical signaling happens. Then if you remember the previous slide, the penetration go deep, and then it disturbs the photosynthetic mechanism. So these kind of things happen. I have given here one example which is well studied globally. It's an example of species called Polysiphonia. Those algae specialization people, I'm sure, must have seen it because it's one of the model organism, Polysiphonia, and you can see its a photo is from our own farm where how much Polysiphonia is growing on Kappa ficus, you can see. And then scanning electron microscope also I have given where people have shown how it has penetrated deep inside the tissue. So if you see a mechanism of action, there are 5 different mechanism of actions described. So first one is, let us see one by one. First one is infestation period, where as I explained you little while ago, the spore comes and it attaches, and it penetrates through the surface. Then a second stage where the spore germinates and forms the body. They are basically a gametophytic plants. So they produce some kind of gametophytic plant, and from which again spores come through in the sporangia. And then that's how they complete their life cycle. I'm not very sure, but Polysiphonia is a classic life cycle. It shows a classic life cycle, and the life cycle pattern also is known after its name, Polysiphonia type of life cycle. So it has 3 stages, male, female is a gametophyte, then tetrasporophyte and cystocarp. So basically, when infestation happens, they basically try to start their life cycle so that they can grow profusely and fast. So this life cycle takes place, and then a stage comes where all the life cycle stages very firmly attaches and form a community where you cannot differentiate between the individual. And they, as I said, both gametophytes and sporophytes are there. And that's how they spread very fast. I would compare this Polysiphonia infestation with the aphids. I don't know how many of you are aware of aphid infestation in higher plants, So the aphids, they have very interesting thing that they also can grow parthenocarpically, and they don't need mating and all. And they basically grow enormously. Same way this Polysiphonia grows. And then understanding and establishment of epiphyte on seaweed surface is very, very critical, because if you can imagine there is a big farm where this infestation happened, and it takes hardly a couple of days, 2 or 3 days or a week's time to wipe out the entire crop. That's how they grow very fast. And that's why this is very important, so that some mitigation or some protective measures can be taken up. I'm going to show you 2 slides. They are mostly a pictorial one. There is no description, just to show you what all kind of epiphytes are there. You see here, all types of epiphytes are there, from brown algae to red algae to green algae on various plants. So basically, there is a lot of diversity. This is another slide where I have given epifaunal diversity. Here also you can see a lot of epifauna is associated. You also have to understand this is their natural home. Seaweeds, they provide a habitat, and for all these people, all these organisms to reproduce, they act as a nursery, breeding ground and protective cover. So that's how they come. Now let us go to second part of my talk is about diseases. There are several diseases. If you see the literature, at least 50 types of diseases of various seaweeds are reported. I'm going to take example of only one because that is the one which is most studied. Okay, it's called ice disease. In next slide, I'm going to give you an example from India. This basically, what happens, a natural assemblage has reported lot of variation. As I said, they provide breeding ground and things like that. So naturally, there is more diversity. Accordingly, diversity in pathogens or bacteria or whatever disease-causing organisms is more in case of naturally growing seaweeds. But at the same time, although diversity is less in monospecific or farmed species, the extent is always more. Like if you see in the nature, one seaweed, another neighboring seaweed is not necessary of the same species or taxa. It can be different species, it can be different group. But in case of cultivation, all the plants in a hectare or many hectares, all are same. So even if diversity of disease causing or whatever the problem causing pest is less, but there is a more area available for them to infect or infest. Earlier, people were thinking that particular organisms cause particular symptom or disease. Let's take an example of some bacteria. So this particular bacteria causes this particular symptom or disease. And scientists or professionals were thinking that's true. It's not true. Lot of new research has come, and now they call it pathobiome, or this is a group of organisms. So one pathogen, one disease concept is not true. It is shown that always there is a diversity, or there are more organisms who basically act together to have a cascading effect. Okay, so a lot of studies are now undertaken how this microbiome composition changes area wise, species wise. All this is done, and that is why understanding disease is very important, and understanding through only one pathogen is not going to help, or we cannot bring out the solutions. We have to understand the role of each organism and how cumulatively they basically kill your seaweed or any plant is more important. This is an example of one of the largest mass mortality event happened in the country. Many of you are aware that commercial cultivation is happening in Tamil Nadu, and 2013-14, at that time I was posted at our marine algae research station. This has happened, and literally this has happened in a day, a 3 or 4 days period. Entire crop was dead. We investigated. Some of our researchers have studied why this has happened. We have come to the conclusion that I have given some data. You see, that was the time when there was a maximum crop. That was the peak season for peak time for the country to have maximum number of rafts floating. And what has happened is, you can see below a small table I have given. We have given only two parameters. One is air temperature and average wind velocity for a week or so. You can see that temperature has not changed much, but what had changed is wind velocity. Very sudden drop. Wind velocity has gone to half of its existing wind velocity on particular day. Now let me explain you. This is a raft type of cultivation. Raft creates a small ecosystem because this raft is basically covered with a fish net below to avoid grazing. So it's a small square frame in which lot of plants are growing. Now you also have to understand each crop has a life cycle, or a farming cycle. In this algae case, it is 45 days. So farmers in a particular village start seeding his rafts, and on 45th day it becomes mature. So many farmers in an area seeded couple of rafts on that particular day, and all together in that village, all the rafts are of same age. So when they reach to the maturity, they obviously need lot of nutrients, nutrient flow or dissolved oxygen, things like that for its photosynthesis and growth. Now you have to understand that this crop has come to a maturity. They are fully grown, and all day, every day, basically because of wind and tide, there is a nutrient exchange, water movement. And imagine a wind is not blowing in normal range and it has stopped. What will happen? They are going to starve. And the same way, basically these plants could not get adequate nutrients or dissolved oxygen, and already there were bacteria on their surface, and plant has its own immunity, and they were fighting. Plants were fighting with bacteria every day, but on that particular day their immunity has gone down because they are not healthy, they are not getting dissolved oxygen, they are not getting adequate nutrient. And what happened? These particular complexes, I have given two complexes, they are known to cause eye-size disease. They basically started growing on this and kill the entire crop. That's how is the story. Now let us come to my last part of component is a grazing. As you all know, it's a food chain. and seaweeds or macroalgae or algae, they are basically at the bottom of the food chain, and they provide lot of nutrition and everything what is required for fishes to grow, and they are delicious, and lot of fishes they come and graze this. My colleague Dr. Ganeshan has carried out one very interesting study in 2006, beginning of 2006. He basically planted some 13 types of seaweeds at one location called Crucide Island. Many of you might have visited Tamil Nadu. There are a chain of islands in Gulf of Mannar, and nearest one is Crucide in a Mandapam group. There he has planted 13 types of seaweeds, and he and his team visited every day, to see which are the seaweeds preferentially grazed. And suppose if particular seaweed is grazed, he will put again the same seaweed in the same place and go to the next day and see what happened to that particular species. Like that, he has conducted the experiment for the period of 1 month, and then he showed how seaweeds are preferentially grazed or preferentially taken up, and that's how one can understand that the fishes also has liking and disliking. I'm going to come next slide. I'll tell why disliking and liking also. So basically, you again can visualize a commercial farm, and these are the right hand side. You can see parrotfish, surgeonfish, rabbitfish, etc. sea urchin. They are the biggest grazers. What they do basically, they go in these fishes. They come in a school. They call fish school because they come in large number. They come and graze your farm. So studies have shown that up to 50% of biomass is also grazed by herbivorous fishes in particular area. Such type of observations are reported from Indonesia, Malaysia, and Philippines, where bigger farms are available. Also, there are some benthic one, not fishes, especially crabs and sea urchin. They also graze on it. And then, as I said, our study also had shown preferential grazing. And some of the photos at the bottom I have given, you can see the photosynthetic tissue is totally removed. So fish also, they don't eat the entire thallus. They basically take a nutritive part or probably a palatable one, very easy to digest or chew. And you can see that how they have nibbled and how they have grazed these seaweeds. There is very interesting story which I must tell you, that United Nations in early 60s has started two big experiments. One is in India and one is in I think in Philippines, and this has come through Commonwealth country program and Bay of Bengal. Some of you might be aware, it's an intra-governmental organization located at Chennai. So three of these funding agencies came together, and they have selected a place called Vedalai in Tamil Nadu, near to Mandapam, and they have started Gracilaria cultivation, and similarly they have started cultivation of Gracilaria in Philippines also. In India, what happened, they selected Gracilaria edulis. This is the species, very well known and naturally exploited for commercial production of agar. Our institute has developed a technology, and this technology these guys have taken and they adopted this village, and they went and told women fisher folk that you please do it, and whatever biomass you produce, we will process it and we teach you how one can earn livelihood. And this program has started with very fanfare. Lot of publicity is given at international level also at both the countries. Unfortunately, all the program was going well for some time. Unfortunately, one day all the entire farm was wiped out by these grazers, and the entire crop has failed. And that was the first lesson country got, that these fishes can basically damage your crop. And we do also encounter a lot of grazing. Very recently, my colleagues reported that.
Dr. Vaibhav, you have another 15 minutes.
Yeah, I think I have only 2-3 slides now, madam. So
okay.
Basically, this is how these grazers they affect the commercial activities. as I said that there is basically liking and disliking. So there are a lot of chemicals involved, and the seaweeds, they produce these chemicals in response to grazers, epiphytes, or diseases, and lot of diversities of chemicals is reported. I have given few of them, and basically because of this only some seaweeds grazers don't touch, and some seaweeds, they produce it as an interaction, as I said, between the epiphytes, and these chemically mediated interactions or secondary metabolites are not much studied, and there is lot of scope to study this. Let me come to very interesting part. This is an analysis we have done for the first time. Although some sort of science is available, commercial losses are not available, or economics is not available. This is the first detailed economics we have given in one of our papers, where we have shown that if these epiphytes, diseases, or grazers come, what happens? We have taken an example of 5 seaweeds. I'm not going to read those numbers. Those are available for investigation if anybody wants to do it. Only thing is, all these have a lot of commercial implication, and these commercial implication are going to show in terms of socio-economic consequences. As you might be knowing, some of the communities are totally depending on this activity, and imagine a scenario where the entire crop lost because of epiphytes or whatever the reason may be. So one has to really do very sincere effort, and studies are required, and also multi-sectoral studies are required, not only from one angle. And this will basically help to a policymakers. We have been talking in so many forums and telling government that please bring seaweed farming also under the ambit of agriculture, and there is a requirement of kind of policy matters where this can be also protected. Now, when somebody talks about these losses and problems, biosecurity measures are very important. Risk assessment is also important. So we have proposed this scheme where one has to really watch for farm. Every day one has to go and check, and meticulous diary or meticulous recording has to be there. A nutrient management, or some physical control. Even before starting a farm, one can select good seed. It starts from there. Good seed, healthy seeds with disease-free or epiphyte-free seeds should be there. One can also bring various crop rotation patterns. One need not have to cultivate only one species. One can take one crop of one species, another crop for another species. Such measures are available for higher plants. We also propose that this kind of integrated pest control approach is required, and this information is also required for taking decisions very quickly to minimize the loss. I think last part I want to touch upon omics approach. Many of the young researchers might be knowing, everybody talks these days about metabolomics, transcriptomics, proteomics. So these kind of studies, although people are doing, most of the studies are of different attributes. There are no much studies on how this omics approach one can use to understand phenomenon of epiphytism or diseases. I think only 2 studies are available, as I have shown, pyropia or porphyra, where people have seen culturing this without bacteria, pathogenic bacteria, with pathogenic bacteria, and they have shown the metabolites related to growth, energy, storage, etc. They altered when you culture them separately, when you culture them together. It's a very preliminary study, but it shows that there is a lot of scope. And then there is another study where basically transcriptomics level analysis is done for Gracilaria changai, where they have used some agarolitic bacterium and also use agarase enzyme. And then they shown that whatever expression of gene, either upregulated or downregulated, that changes based on inoculating this bacteria, either bacteria or agarase enzyme, because both of them do the same work. and that's how one transcriptomics only one transcriptomics study is available. What I wanted to say here is there is a huge scope of taking up some of these studies as the PhD topic. I think this is the last slide, madam. I'm concluding here. I'm not going to read. Let me only tell you very few things. There is extreme lack of information in this domain, and one has to really work hard to compile and bring it. It has related to livelihood and economics in the coastal rural setting, so one needs to do it. Most of the information, whatever is available, is from natural habitat and not much in the commercial farms. There are total lack of taxonomic manuals, keys, identification, and global level no database is available. So one has to really do it. So many organisms have different life cycle stages, and these life cycle stages also one need to study. There is lot of scope of using some kind of pre-treatments. One of them are seaweed sap itself or seaweed extract itself, which had shown some beneficial effect. One can basically do it. Crop breeding program, most of the programs are talking about yield and growth. There is a need to basically develop disease resistant varieties. Bioengineering and all also is another area which is open for research, and protocols need to be developed where one can actually assess in the field, because most of the time you take a sample, take it to laboratory and study, time goes, and then one can basically talk about developing protocols which can ascertain the damage or study the stage at which it is in the field itself. The entire talk, when I talked in my lecture, every negative aspect I talk. I don't want to stop my lecture with negative aspect. There is one positive aspect of these epiphytes and associated things are they also basically protect the plant or seaweed from pathogenic, kind of or fouling kind of organisms, and one also needs to consider. I think with this comment, I stop here. I thank my entire team, especially students, Durga, Kapil, Donia, and my colleagues also, all who have involved in this study and this program. Thank you very much. I am very happy to take questions.
Thank you. The session is open for questions. And I think there are some questions. You can read this in chats. How volatile compounds are responsible for bioactivity of seaweeds?
Yeah, volatile compounds are very much important. Volatile compounds is another area of research. But they all act when seaweed comes out of the water. They don't act when they are inside the water. That's what is observation. But most of the time, some of the seaweeds produce these kind of compounds and keep the grazers away during low tide. That's what is my information.
There are 3 questions from Joel Fernandez. What is the proxy you use to check for immunity?
Okay. One of the very important parameter for checking immunity is antioxidant activity. Okay. So there are protocols. There are, at least in my lab, we have developed 3 protocols, and we just see how antioxidant behavior of that particular alga is there. Antioxidant properties is the one. I can send papers. And
second question is, it is interesting that you suggested the microbiome is important in pathogen mediated susceptibility of the seaweed. Yes. Are you currently assessing possible communities within the microbiome that are important to keep the pathogen in check?
One of my student, Donia, she is working on this, but we are not looking it in terms of diseases. We are looking it in terms of what is the microbiome. That's what we are doing. We are not doing what happens to microbiome after infestation or infection. We are just checking what is the microbiome present. See, we all work on seaweed cultivation. We are interested to know what happens during cultivation. So what we did in that study is we have collected the alga from nature and we studied its microbiome. Then we put it in cultivation practice, and we see what is the microbiome. We just wanted to know which are the organisms which are really remain there. That means they are required for its natural growth, and that's what we are interested in.
And the third question is, he loved your observation about the role of wind in disease progression. Is this a general phenomenon with different germplasm of seaweeds?
No, I will tell you, this is very peculiar case, and it was possible because at that time the production was at its peak. It doesn't happen in nature. Okay, nature takes care of it. We have artificially put a raft, and we concluded that that was the only thing changed on that particular day, and it might have triggered, a growth of pathogenic bacteria. That's what is our assumption.
I have one, I don't know, it is not a question, but I just leave it to you as a sentence.
Yes.
In the members of Rhodophyta, in general, when we were taught, or when we read the characters of Rhodophyta, we speak about the pit connections. That is primary pit connection, secondary pit connection.
Yeah.
And there is always a note that there is a pit connection can be formed with the parasite also. So how do you relate this?
Madam, I think it's, again, as you said, it's my assumption, and it's I don't think it is proved or not. I feel when pathogen infect any system, then they don't remain two parts. They become together, So the pathogen develops this pit connection, or whatever you say, is because it has to take the nutrients and all those things. And that's how, I don't think then they remain two systems. They become one system. Some of the genes also are transferred. There are studies where it is shown that pathogen gene also are transferred into a host, and host genes also are transferred into pathogen. That's what is said. So I feel.
So since you are working in this line, you may take up this work and study this. I think it will be a good observation. I think there is one more question. How can seaweeds be helpful in sandponics?
Sandponics, I don't think, because you see, they grow inside the water. Probably your halophytic plant or something like that, they can help because they are inside the water, and they need not have to grow only on sand. They rather grow on rocky surface luxuriantly.
I see one more question here. Any control measure to minimize or to eradicate the epiphytic disease during the cultivation of seaweeds in commercial farms?
See, the only measure is not allowing it to come. It's like taking a precaution. Precaution is the only solution. Once it comes, the they have to basically destroy the entire farm. They have to harvest it, dry it, and they should not take the same species again for at least 1 cycle or 2 cycles. That's the recommendation, international recommendation. So best is prevention.
I will take up the last question. Will there be possibility of any uptake of xenobiotic or any other toxic chemical or particles by the seaweeds? Would it have effect on human after consumption of seaweeds or act as vector for marine food chain?
Definitely, as a biologist, we all have to understand that seaweed also is a system. It takes whatever is there. Only thing is how much it bioaccumulates is the study, and it will definitely have an implication on human health. But there is a catch here. If you eat seaweed as such raw, it will have that effect. If you take out the products, then in process, everything goes out. So if you eat seaweed as a raw or seaweed directly, it will accumulate and will have. If not, it won't have.
So with this, I thank you very much for your interesting topic, something different. And I'm sure after the pandemic, we'll have a chance to meet in person. So
Yeah.
thank you very much. And once again, on behalf of my own behalf and my students, colleagues, and department, university, thank you very much for, there may be some errors from our side, but in spite of that, you have cooperated very well. Thank you once again.
Madam, let me also take a moment. I would like to thank all of you. There are no errors. We all are colleagues. I am very much thankful to you, head of the department, university, and I'm thankful to my director also for always encouraging us to give our knowledge to betterment of society. Thank you so much.
Thank you. Siddhi, you can go with the next session.
Yes, ma'am.
Yeah. I request Professor Janardhanam to take over the next session.
Yeah. Thank you, sir.
Thank you.
And this is the last session. And so Dean Professor Vijaya Kerkar, Head of the Department Professor Krishnan, Organizing Secretary Dr. Siddhi Jalmi, and all other colleagues and students and participants. And before I introduce the speaker, let me directly thank the speaker, Dr. Ganesan, for accepting our invitation and being here with us today. And Dr. Ganesan is going to talk about conserving biodiversity beyond protected areas. We all know that ultimately, whatever we have been seeing, signal processes or molecular processes or bioprospecting, everything, ultimately, we are also talking about parallelly about losing our biodiversity. Though we have laws, everything, but we think that once we declare biosphere reserves, wildlife sanctuaries, that will take care of the conservation. It is not true. That is what Dr. Ganesan is going to tell today. And before he starts, I would like to introduce Dr. Rangayan Ganesan, who is a fellow, Suri Sehgal Center for Biodiversity and Conservation in Ashoka Trust for Research in Ecology and Environment, which is very popularly known as, well known as ATRI throughout and outside the country in Bangalore. And Dr. Ganesan did his graduation, post-graduation, MPhil, PhD, all from Madras University. And he is a member of so many bodies, including Indian Association for Angiosperm Taxonomy, Systematic Association UK, International Society for Tropical Ecology, India, Association for Tropical Biology and Conservation, ATBC, USA, and the International Society for Tropical Foresters, USA. And his specialization, when I say he specializes in different aspects and bring it to a common platform. He's a plant taxonomist, he's a plant ecologist, he works on livelihood issues, he works on conservation, and he puts all of them together. Ultimately, whatever we do should be useful to the society, and that is the probably mandate of Suri Sehgal Center for Biodiversity and Conservation, and he puts very nicely all of them together. And his research also includes long-term monitoring of biodiversity changes through long-term monitoring of vegetation in Western Ghats, improving and managing green cover of urban landscapes, also disseminate knowledge about diversity and functional aspects of biodiversity, and help in public and policy making to influence biodiversity conservation. And having known Dr. Ganesan for more than 3.5 decades as a friend, colleague, we discuss a lot about a lot of these issues. He has not given his full biodata, which he is so down to earth. So I do, though I know a lot of things about Dr. Ganesan, I would restrict myself here. How do I know? I told you it's more than 3.5 decades of our association. We were working together in Botanical Survey of India. Then he also joined Salim Ali School of Ecology in Pondicherry University. Then it's a long journey in his association with various universities and institutes across the globe is also there. And regarding the title of the talk today, Conserve Biodiversity Beyond Protected Areas, is 2 latest papers. One, Inclusive Conservation Using Long-Term Integration of Natural and Social Science Research, one, and Rhododendron Conservation in Himalayas. Would people who want to know more, probably you can look for these papers. Being latest, I am just telling only those 2. Though he has worked on, Phyllanthus emblica, Phyllanthus Indo-fishery as models in Bilgiri-Rangan hills and all. And I will not go beyond this. And thank you, Dr. Ganesan, once again. And we are all ears for you. Stage is yours.
Thank you, Dr. Janardhanam, for such a very lucid introduction. And really, I'm very much humbled and honored to get such a kind of introduction from a kind of Bhishma, Bidamaga, whatever we call, because our relationship was that always. I used to learn a lot from Dr. Janardhanam, and I'll keep learning many things from him. And also I thank Dr. Krishnan for introducing me to Goa University colleagues about the expertise I have, which I can share it with all of you. And also I thank Dr. Jalmi for her patience, despite of several reminders. I was not able to be on time in responding to her mails, but due to some other commitments, I was not able to do. I'm sorry. So I thank you all for this wonderful opportunity, and also I thank the audience. Hope my presentation or the lecture will be useful for all of you. Hope my slide is visible to all of you.
Yes, sir, it is visible.
Okay.
Only thing, we can make it full screen if possible.
Yeah, I made it full screen. Yeah. I made it as a full screen mode. Still,
it's set to appear like that.
Okay. Okay. So my, as Dr. Janardhanam introduced, it's about conservation. Generally, we talk about conservation, and immediately protected areas comes into our mind. Animals comes into our mind, especially in Indian condition, tigers, elephants comes into our mind. So a few months back, we might have, and most of us might have celebrated or taken participation in Environmental Day, World Environment Day. On that day, it has been declared by UN as a decade for ecosystem restoration. And we were all very, very happy, and then we all actively participated with various interests. And above all that, all of us might have been, most of us might have been involved in planting trees, thinking that restoring our green environment is what going to take our world in a good healthy condition. And also when we go through some of our social media or publications or internet, we get to know this kind of information also. Sometimes it will be very depressing to see. So 3 days back, when I opened my computer, I got a message. The first one you may be seeing, National Geographic. As part of the newsletter, I get to know the doomsday clock shows threats to Earth. And when I get into that, there was a movement called Turn Back the Clock. And again, I went back into UN site where I saw a warning, 9 more years to restore the planet. And these are all telling us that we are running against the time. And every one of us know that there are lots of issues regarding our environment, especially biodiversity. So how do we work? But generally, we pass the bug on to government agencies, largely Forest Department, or NGOs who are more concerned about environmental conservation or biodiversity conservation. And then few of you means audience will be personally interested in participating in various activities related to improving the green cover. So even I also was part of it, and I was called by Bharathidasan University, Tiruchirappalli, to talk about restoration aspect. And I also was critical, do only the planting trees are going to restore our environment? Because this is a science I am also practicing as part of my career in addition to plant systematics. So, and then I saw many references and online social media titles like this, the surprising downsides to planting trillions of trees, because there was a movement that says that trillions of trees we have to plant to protect our environment. And they're also following that, there were also criticism, more of cynical criticism, many tree plant. And Dr. Shankar Raman from NCF, he wrote the following thing, like why frenzied tree planting is no answer to ecological restoration. I was very impressed with it, because saying appropriate species has to be planted in appropriate habitats. So overall, the message is that tree planting alone is not going to restore whatever the damage we have carried out so far. And the kind of whatever the tree we are planting is not also going to take care, because first thing is trees are not really going to salvage whatever the damage we did. Second thing is that not going for appropriate trees. And then because I heard that many of you are master's students, I thought of sharing some basic information. So up to 2019, where India was there about biodiversity conservation, it's basically about declaring protected areas. So you may be seeing the numbers. So right and left, we keep declaring protected areas and proposing government also to bring in more protected areas. But we also know that our country is very thickly populated, and we have very limited area under forest cover. And historically, we also share the forest and the forest denizens with the human life. So it is like more of co-living with the wildlife in the forest. So is it really going to be more effective? So with that, when I was going through the literature, I get to know that.
Sir, are you changing the slides?
Yes, I've changed it.
No, it's stuck up with the first slide only.
Oh, I'm sorry then. So otherwise, can you play the slides from your system?
Can you send it?
I sent it to Dr. Jalmi.
Okay.
Sir, I'll see. I'll project it here.
Yeah.
I'm sorry, really. I don't know. A week back also the same thing happened. Maybe with my computer, some delay.
That's fine. We'll get it back.
Yeah. So shall I wait or keep continuing?
I think just a minute so that she will.
Okay.
Or yes.
Maybe I'll unshare.
Yeah.
Stop presenting.
Yeah, stop presenting. You may have to say probably when she presents next and all.
Yes, sure.
Like our old slide changing.
Thank you so much. So based on my last experience, I thought I should go for the plan B. That's the reason I sent it to Dr. Chalmi.
Dr. Siddhi, you can also send it to me if you want. I can change it.
Oh, no problem. Yeah, I will do it. I'll do it right away.
Okay, sir. I will just give me 2 minutes.
Yeah.
Sure.
Ganesan sir, if you have readily, you can also send it to me. Jana.
Yeah. I'm going to do that. So shall I send it to your Gmail, Dr. Janardhan?
No, jana@unigoa.ac.in. Jana, just now I have sent a mail to you.
Yeah.
From there you can respond.
I forwarded the message that I have sent it to Dr. Jalmi.
Okay, yeah. It takes just 1 minute, you don't mind.
Sir, I have sent you the file, but it is
okay.
getting loaded here. Yeah.
I got it. Dr. Ganesan, you don't worry. I got it.
Thank you. I will try to rectify it in future for this.
No, maybe it is too big a file. 64 MB. That may be the reason why it is not changing. Doesn't matter. I will try. It's still downloading. It will take just a minute, I think.
Maybe I should have converted it to PDF and sent it.
No issue. I will just give you 1 minute. Only 1 minute. It's just loading. It's a big one, no? Maybe that is one of the reasons why it was hanging.
Okay. I'll try to sort it out. Okay, yeah, I'm seeing the slide now.
You are seeing?
Yes.
I didn't share it. No, who is sharing this now?
I think Dr. Jalmi is presenting it.
Sir, I am sharing the slides.
Okay, fine. Now you carry on.
Okay.
Yes,
shall I start?
Yes.
Yeah.
Oh, yeah, it's visible now. Thank you.
So so when we see that,
Sir, can you tell me the slide number? Where should I?
Please move on. 4th slide, I think. 4th slide. Yeah, next slide, please. Yeah, next slide. Yeah, thank you. Yeah, this is the one. So when we see the literature, it shows that declaring protected areas alone are not going to solve the issue or going to help in conserving the biodiversity. Of course, species-specific protected areas like tigers or elephant or dugong. So it is not going to be very helpful. Neither the species and its habitat is going to be protected for a long or even during the short time. And they are suggesting, based on the research experience, the studies say that we have to find some stepping stones, that is some semi-wilderness areas around the particular areas also have to be given formal protection. Formal protection in the sense not by fencing and penalizing the community. So, but they can act as a stepping stone from one protected area to another protected area. Corridors have to be established in such a way seeds of the plants could move, animals also can move from one protected area to another protected area. Second slide, next slide, please. So the question is now, just by making protected areas, that is making Wildlife Act more and more stringent, and then making more and more amendments, can that really going to help conserving the biodiversity in India, like landscapes? So it is a big question. In number of protected areas keep increasing, but still we are very much concerned that many of the biodiversity are not protected. Even the protected areas, some of the protected areas supposed to be having tigers, even there also the tiger numbers have come down because there are various other ecological issues connected with habitat management really interferes in the focal species based protected area conservation measures. And also lots of uproar from the people who are dependent on this forest for their livelihoods, generations and generations. So it becoming a kind of overall the protected area based conservation approach is called as fencing and penalizing. Next slide, please. So with that, I would like to share my research experience, and of course, many of you can also think of such kind of research that you can carry out from the landscape in which you are all going to be there. So, for example, just southwest of Chennai, there are a group of forests. They are more of outskirts of Eastern Ghats, called as Thiruvannamalai, Jinji Hills, and Tirupattur Hills. So quite a lot of biodiversity is there. But generally, we botanists think, oh, these are all small hillocks or small mountains. But to my surprise, one of my colleagues, Dr. Narasimhan from Madras Christian College, he invited me to be part of it to help in identifying some plants as well as doing some quantitative studies. So surprisingly, you may be seeing that on the right side, Dr. Narasimhan and his team collected a plant, but they suspected that it may not be the usual Annonaceae plant that has been universally identified. And then after 2-3 years of toil, they were able to identify it as an undescribed species. And that was quite a surprise for everyone, but not for botanists like us, but we always expect that there are lots of such hidden treasures in these kinds of isolated mountains and forests. And you can see that the small, in the left side picture, you'll be seeing that immediately lots of fallow fields and paddy fields and water bodies, rivers, and then again you see the mountains, small mountains. So these are all having lots of hidden treasures. Next slide, please. And this is another endemic plant, that is Musanda tomentosa. You can see that there is a shrub right in the middle of huge boulders. So the hillock is nothing but it's a kind of heap of huge boulders. And this is representing the Deccan landscape, that is entire South India, especially the Deccan landscape. And there you see this Musanda tomentosa. Generally, Musanda, we all have lots of cultivated plants. They'll be having the white calyx, which adds more attraction to the pollinators, whereas in this plant, they don't have that kind of white leafy structure, that is enlargement of the calyx. So this is an endemic plant, and in fact, its type locality is mentioned in the literature as hills. Maybe, I don't know, from this area, the original collector has been collected it. Next slide, please. And you all might have heard of Hilgardia papulifolia. It's supposed to be along the coastal forest, Coromandel coast forest, but there are a few trees right in the middle of these big, huge boulders. Next slide. And we may think as a botanist, only plants are there. Of course, I also take some interest in taking pictures of curious animals when I do botanizing this kind of habitats. So to my surprise, just in 2 trips, I got these 2 animals. One on the right side, you'll be very surprised to see that it's called golden gecko. But for any one of us, the gecko will be looking like the gecko that we see in our houses. But if you see the toes, you may be able to see that how different this gecko, and the call also was very different. So I took the picture and shared it with my friends, and then immediately they said that is a golden gecko, very, very rare. And I looked into the literature and found that it was described from Tirupattur Hills, just crow flight distance, maybe 25-30 kilometers from the place in which I took this picture. And when I was going along with my friends, that was a rainy season, I also saw this snake and took this picture, and then saw that it's a shield-tail. And shield-tail snakes are again endemic to Deccan Plateau. And each hillock supposed to have a species, it seems, according to Dr. Whittaker, the snake expert. So, next slide, please. So again, in Madurai, the Crotalaria digitata are supposed to be extinct, and in IUCN also it has been marked as probably extinct. To our surprise, when we are doing a quantitative study in different region, this species appeared, and then one of my colleague Kotamuthu identified this as a species that is reported as extinct. And then, and also there was a wrong identification that led the species to be recorded from Cool, maybe some 600 kilometers away from Madurai. So these all made me to feel that the habitat it's a huge boulder, again Deccan hillock-like boulder, surrounded by cashew plantations and farmlands. So it is. I would say that it is rarer, and it requires more concern than the tiger to be conserved. That only few hundred plants are there in that hillock, and following that, it's more than 10-15 years. Still, we keep searching in different parts of this ideal habitat. We are not able to see this species. So next slide, please. And these habitats are not part of any protected area. That is what I would like to show, that there are biodiversity right in the middle of human-dominated landscapes, and they have gone unnoticed, or they have given least priority for conservation. But if you ask anyone, immediately they will say that declare it as a protected area. But my understanding, and many of the researchers, many of you will also understand, it is not practically possible to conserve each and every square inch or square meter of the forest of India as a protected area, fence it and penalize the people. So protected areas is a kind of generally among the conservation biologists, but but over the 30 to 50 years time period, it has been realized that protected areas are not the only solution to protect many of our endemic plants and their habitat. One thing is that it is not possible. Second thing is that ecologically, those protected areas cannot really be viable solution because of the genetic exchanges between these endemic plants and endemic animals. If the protected areas are isolated, that is why they say that functionally connected networks should be maintained and restored based on landscape connectivity perspective. So next slide, please. Yeah. So I would like, generally when conservation comes, we talk about rare animals, threatened animals, rare plants, threatened plants, medicinal plants. So that is a kind of general approach. But I feel that to include communities, to include the people also as part of the conservation, and more a kind of middle step, we have to take a middle position to conserve the biodiversity in our country, in addition to protect area, can be centered around crop wild relatives and wild edible plants. So this is my recent experience. Of course, many of you know that quite a lot of research has been carried out across India as well as globally, that crop wild relatives are very, very important plant. And the teachers might have taught you all about the importance of crop wild relatives. I'll come on the next slide. So the thing is that crop wild relatives, and you may be seeing that red color that highlights the number of crop wild relatives, especially in the part of India and many part of the world also. It is, I would say that we in India may be having more than 150 crop wild relatives. Next slide, please. Yeah. And you might have heard of, as part of your biogeography, phytogeography, you might have heard of Wavilian centers of biodiversity or crop diversity. So India is one of the center, and India has various sub-centers also for Wavilian crop diversity. And India has very, very long history of human presence. And then if we see that, really we'll be surprised to see the kind of bubbling biodiversity and how the human with the wildlife really practically happening in India compared to any other part of the world. Because we respect each and every plant and then they are important bioresource for us. And not only plants but also animals. So in this kind of matrix, and that is human dominated landscape with protected areas and the habitats which are not really under the formal protection measures, or with bubbling with biodiversity, and how do we conserve them. So I thought of bringing in a kind of different kind of approach where the approach could be amenable to the people who are very strictly for protecting the biodiversity under protected area management regimes, as well as some of them would like to take the middle path through which they also want to have co-inhabitation of people as well as wildlife in such a way that we can let the plants and animals evolve along with the human's presence in such a way that biodiversity remains as biodiversity. Next slide, please. And crop wild relatives, and most of you will be knowing, but even then I thought of unpacking it a little more. So we have pseudo cereals. One is called amaranth. Amaranth, it's a dicotyledonous plant, and that seed is being extracted and used on par with cereals. And then, of course, you know that there are lots of cereals, millets, and pulses. Several pulses, of course. So Western Ghats is known for supporting many of the crop wild relatives of our PGNP, chickpeas, and of course many oil seeds. Of course, gingerly oil, gingerly seed is the wild crop wild relatives of gingerly seed is one of the important things from Western Ghats. And we also know that India has quite a diverse banana crop wild relatives, Musa. Next slide, please. So millets, you can see that our country, over the 2000, some people say 2000 years, some people say 5000 years, but whatever the number of years, India was a hotbed to domesticate many crop plants, and the barnyard millet, that is Echinoclova, is one such. Next slide, please. And Kodo millet is yet another species that has been domesticated from India. It's found still in the wilder form. Next slide, please. And if we see that, what is the importance of crop wild relatives? Generally, we have, they say that, we have, say, not more than 15 crops, and then with that, it should be sufficient enough for us. But what are the importance of having crop wild relatives still in situ? In situ in the sense where they are mostly in the wilder form. So generally we talk about now food security, and above all that we also talk about nutrient security, because food security, we feel that we have achieved that, but the kind of food we are having are not having the essential nutrients. So we talk about nutritional security as the next step. And also by having the crop wild relatives in a safer condition, we can also get through climate change related stresses, pests and pathogens. And globally, these crop wild relatives are considered as weeds. But you can see that the monetary value of these crop wild relatives, which are contributing to our human kind. Next slide, please. Next slide. Yeah. So in addition to crop wild relatives, I also would like to add wild edible plants as part of the conservation strategies outside the protected areas in such a way that they will be part of the stepping stones and corridors that connects to protected areas. So many wild edible fruits, which we generally think, oh, they are the food for indigenous community, or they are the food for very poor people or marginalized community. But nutritionally, they are very, very rich. And that is where I talked about, that is what I mentioned about nutritional security. For example, this Karvandia fruit, I think I took the photograph from the outskirt of the Goa, where the indigenous community women were selling it along the roadside right in the Goa. And the leaf, you can see that she made a packet or a pouch out of the Butea monosperma alas tree. And then you can see in the middle, Beccaria cutalensis, especially for the lactating women, the indigenous community recommends these fruits to be eaten, and malnourished children also supposed to eat this fruit. And Elaeagnus cologa, it's another wild fruit. So we can, next slide please, we can keep on listing quite a lot of wild edibles. And especially if you go to Northeast, many other markets, in addition to usual onions and potatoes, brinjals, you will see quite a diverse, especially in the wiggly sandies. You will see quite a diverse plants that comes from the neighborhood, comes from the community forest, that comes from the protected areas. So here, so you'll be seeing what is so important, why we are so concerned about crop wild relatives and wild edible plants. Because when we talk about conservation, we have very good Wildlife Act, we have very good Biodiversity Act also to conserve the wild plants. And similarly, Agriculture Department, our Agriculture Department policies are also protect many other crop varieties. And then we have lots of subsidies and lots of efforts to conserve the crops. But there is a crack in between these wild animals or wild plant resources and the agricultural crop resources. That is where neither the agriculture related policy nor the conservation related policy or activities can really conserve these crop wild relatives and wild edible plants. And this is what should be our concern for the future conservation activities. Next slide, please. So if we see for example, I have a project in Northeastern region where we would like to map the distribution of wild biological resources in the entire Northeastern region, including Himalayan forests, and identify some of the important wild plants that can be used to improve their livelihoods. So you can see any plant in the forest that has quite a lot of value from the edible point of view, as a food point of view, or nutritional security point of view. And you could see the numbers, but you can Google it and find out these numbers. But I was really surprised that a huge number of bananas, huge diversity of bamboos are important part of the crop wild relatives and wild edible plants in the Northeastern region. Of course, in South India also we consume bamboos and bananas, but not the wild bananas. Next slide, please. So the quantity of biological resources sold in the market is really humongous. One cannot imagine. And I would say that more than the grains from the fields that has been sold in the Northeast, these wild plants may be playing a much better role. Or the quantity of wild biological resources sold in the Northeast market are really very significant, not only the quantity, but also taking care of food security, providing nutritional security. In addition to that, the amount of income generation that comes through this trade of wild biological resources or wild edible plants are really mind-boggling for most of us. So just you can see that the data which I show here, it's a very smaller area, Assam, Sikkim. and North Bengal, part of Assam, not the entire Assam. So there are around 258 species we have identified. Of course, I think 1/3 of species we are not able to identify because they are in either they are in foliaceous plants like leaves and stems are sold in the market. So we are taking effort to identify them. And interestingly, you will also get to know that snails and various insects are also sold in the market as part of the wild edible plants and wild edible animal sources. And interestingly, you all will be surprised to know that we collected, one of my colleague, Dr. Arvind Madhyastha, collected the snails from the market, 2-3 places. And he was able to say very confidently, and then he had described at least 7 species of new snails, which are not recorded so far from the scientific reports. So that shows that even before they get identified or properly, we get to know their distribution and their distributional range is get to know, and their population status is known. They are in the market. And similarly, I also expect that at least few plant species are going to be part of that suite of wild edible plants that are sold in the market. And many, I feel few of them could be undescribed, new to science, or they may be having interesting distribution pattern, or maybe very rare distribution, where they are very much restricted to very limited ranges also. The thing is that I would like to highlight, this is not only in the Northeast, this is also happening in South India and other parts of India. But we researchers, we list those species like crop wild relatives or wild germ blossoms. And then we also don't quantify how much they are sold and what is their contribution to the economy of the traders and then households also, because these crop wild relatives play a very, very important role because they are sold in a very, very cheaper price, or sometimes they go to their backyard or they go to their fallow fields or they go to the backyard in the sense the forest, and they harvest those day-to-day requirements, and then they subsidize their family requirements from the point of monetary demands. And also it is they not only food but also they are taking care of health requirements in the form of medicinal plants, and quite a lot of plants are also wild plants are also harvested and sold in the markets, and these are all the plants not coming from the protected area. Again and again I talk about this bioresources, especially crop wild relatives and wild edibles, wild medicinal plant resources are all from the human dominated landscape. So how do we conserve them? Next slide, please. And this part I haven't talked about it. So the thing is that I try to give a kind of projection that the plants which are mostly crop wild relatives and the economically important plant collected from the wildness or the wastelands or the so-called wastelands or the locations around the paddy fields or around the canals, along the roadsides and anywhere. It doesn't mean that our protected areas do not have that, but the thing is that because of fencing and penalizing, we are not supposed to harvest the human, the kind of crop wild relatives or the wild edibles. But what is the status of crop wild relatives and other wild edibles inside the forest? Because our concern is to conserve tiger or any such charismatic animal for which the protected areas are declared. So we try to manipulate the habitat, or we leave the habitat as such, thinking that the habitats are supporting the wild animal for which the protected areas are established. But from our ecological understanding of many of the plants and animals, there are certain levels of modifications in the habitat or manipulation in the habitats are required for the populations to thrive. But according to the Protected Area Management Regulations or Biodiversity Act, we will not be able to do that. So there is no point of hoping that these crop wild relatives and wild edible plants will be conserved inside the protected areas, because they are for a different purpose. and crop wild relatives and wild edible plants require different approaches. I think that different approaches, I mean to say that there is a need of constant manipulation in their habitat. That is what happening, and that is where our community are harvesting the crop wild relatives or wild edible plants or medicinal plants. So, so please move on to the next slide. So so here you can see that on the right side you will be seeing a fruit called Michaelis edulis, and at the bottom most of you or all of you might be very familiar, that is Persea americana. It's an avocado fruit, and we also call it as butterfruit, and it's supposed to have about 40% of the protein. Generally fruits are richer in fibers and sugar, vitamin, amino acids, folic acids, and so many other things. Whereas this avocado fruit very unique in having about 40% of protein. So above that you will be seeing Michaelis edulis. Of course, earlier it was Persea edulis. Because of nomenclature, now it is shifted to a genus called Michaelis edulis. This is a wild fruit. This is also found in the protected area. Also, this is also found in community forests. And these tree species also protected by individuals if they are in their backyards, and agroforestry practices. And the thing is that by as I mentioned here, outside protected areas are primarily managed for reasons other than conservation. So that is how we have many other crop wild relatives and many other wild edible plants. And then here, if we are going to say that it has to be protected and we are going to bring in under protected area regime, community will be offended, community will be affected. Not only their economic benefits will be affected, but their health also will be affected. And that is how we see in many other protected areas, especially tiger-centric conserved areas. People are evicted and they have been given compensations in such a way that they can go outside and practice their farming activities or and their skills, which are mostly used for foraging in the forest, more of hunter-gatherers, but they have been immediately shifted out in the name of tiger conservation, and they are not having any livelihoods because they don't have the skill to do the farming outside the protected areas. Whereas if we could able to provide opportunities by having this kind of micro scale protected areas, but with under the conservation efforts of communities, we will be able to really see a kind of a landscape that is a matrix of landscape that has few protected areas and many community conservation areas, which are the community conservation areas are more connected with crop wild relatives or wild edible plants. Next slide, please. So this is my last slide. So basically, I'm planning to show that how the scheme can be really put in practice. Basically, our overall goal, that is biodiversity conservation at the landscape level, can be achieved. And within that overall goal, we can also conserve crop wild relatives and wild edible plants. So how do we do that? So we know that we have to do crop wild relatives distribution and population genetics data. Of course, researchers, we generated data and we'll be generating data. And of course, all the crop wild relatives and their habitats or all the wild edible plants cannot be prioritized for conservation because of the resource crunch what we have now. So we have to prioritize not only their value as a crop wild plants or wild edible plants, but the kind of threats their habitats are going through. So once it is done, then we have to go for in situ conservation. And in situ conservation, we have to have different kinds of policies. At present, we don't have any such policy that really takes care of in situ conservation of crop wild relatives and wild edible plants. So we have to do that. And then once we are doing it, we also have to monitor it, because this constant interaction between the harvesters and the plant really will have an impact on the population status. Over time, over extraction also can lead to erosion of the population, not only erosion, but also genetic diversity also. So the research community had to work with the communities who are into crop wild relative conservation and wild edible plant conservation. And government agencies also have to take into consideration, because we have National Bureau of Plant Genetic Resources, and they periodically collect crop wild relatives and wild edible plants to keep in their germplasm collections. So when they collect it, they also have to monitor the genetic diversity of these plants. So they all have to, and the kind of agencies who are more concerned about wild germplasm conservation and wild plant genetic resource conservation, and the communities, forest department, research institutes, academicians, all have to come together to conserve the biodiversity outside a protected area. So the ultimate goal will be developing new kinds of policies. So that makes the indigenous community or the farming community or the community that takes care of the in situ conservation with academicians and forest managers and international and national level biodiversity conservation agencies, in such a way that our overall goal, ultimately, as Dr. Janardhan mentioned, that we will be very extensively working on genetic diversity, molecular part of the crops, molecular diversity of the plants. But the in situ interaction by the plants and animals are really very, very important, because that is a cradle in which the plants keep on evolving with the changing environment. And we talk now about climate change, and then plants and animals are also tuning their adaptive measures in such a way that we human are going to ultimate beneficiary. So with this, I would stop here. So I would say that a matrix of landscape that includes protected area and off reserve community-centric conservation measures that involves you and me in conserving our biodiversity will be the ultimate answer for our humankind to live forever. Thank you very much. Once again, I thank you all the Goa University and Dr. Janardhanam, Dr. Krishnan, and Dr. Jalmi, and the participants. Thank you very much.
Thank you, Dr. Ganesan. We will see if any questions are coming, or they need some clarifications, or they would like to supplement with some information. Anything is there?
Sir, you had shown some ferns in the as a bioresources in some Sikkim area. I would like to know more about that, because I had read that Ophioglossom is also consumed in those areas, and now it is endangered because of the overexploitation.
Yes, madam. Thank you. It's very nice to hear that that has got your attraction. Generally, I worked in South India, largely in Western Ghats. When I went to Northeast, that was a surprise, and I also, when I was with the community, they served me this fern shoots. They call it as fiddle fern shoot. That young shoot they harvest. You can see that circinate vernation. So that level, very young, they harvest and cook it. Just they sort it and serve to us with salt. So the thing is that these ferns are playing very, very important role. That is what our studies showed. In quantity as well as quantity by kilograms, as well as monetarily, it is one of the very, very important fern and the plant-based bioresource sold in the northeastern region in many part of it. And these plants, these ferns are harvested largely from the buns in the fields or backyard and many other fallow fields. It was a quite surprise to know that the huge quantity comes from neither from cultivation nor from the forest, but from more of agroforestry systems. And at least 3 species of ferns are harvested, but I am not mastered in identifying the ferns which are in the market, but quite a diverse. Diverse in the sense 3 species, at least from my experience, and huge quantity of ferns are sold. And we are thinking that this could be one of the species as part of organic marketing that is in full-fledged in Sikkim state. So they can, the indigenous community can really substitute their nutritional requirement in addition to monetary benefit. Thank you for that thing which you have highlighted, madam.
Sir, just would like to mention that in Goa also, we eat one fern, acrosticum, so that the young shoots are sold in the market.
Yes, madam. Yeah. A month back I was in Dandeli. That is when I get to know that even in Western Ghats, especially northern part of Western Ghats, some ferns are sold. I collected it, but I have not identified it. But thank you that you have observed. Really, it is very tasty fern shoot preparation and very simple preparation too. Thank you. Yeah, it is an important wild edible plants in Western Ghats too.
Sir, there are a couple of other questions. Mr. Joel Fernandez has asked, was wondering if you have compared the yield of these varieties versus commercial varieties.
Of course.
Also, have you compared the differences in nutrients?
Okay. Yeah, there are two aspects which will be on either end. One is you asked about the quantity. Of course, crop wild relatives and wild edible plants cannot match the crops that we cultivate in the field at present, because we do a lot of crop improvement practices through which we get high yield per unit area. But the second question will be conflicting. I think you might be having an interest to get to know that a lot means really lot a lot from the meaning of benefits from the nutrient point of view. Of course, I will say that a lot doesn't really mean that really they are rich in nutrients and various other benefits. Especially lots of studies have been conducted in the recent past. I think at least there was a study in 2017 where they compared per unit area, per unit of the millets and their nutrient benefits, fiber and other benefits, compared to the kind of cereals we are taking. And definitely there was at least 5 to 10 times high richness of fiber and nutrient content in the millets, especially those are all from the wild resources. At least even if we cultivate millets, those millets are much superior than the crops that we are cultivating. So quantity wise they are lesser, but benefit wise from a nutrient point of view, they are very richer. Hope I have answered my question, Fernandez.
Next one is Mr. Vinayak Kanolkar. What strategies are applied to conserve Orthodextria quartata, a wild variety of rice, which is also seen in Goa?
Okay. Yeah. This is what I was talking to you over the presentation, that many other crop wild relatives are there in the so-called, I will not classify them as wastelands, I won't call them as wastelands. So those are all the places we get many other crop wild relatives, especially the paddies, crop wild relatives or the paddy plants. So how do we conserve them? Because we are doing more and more reclamation of ponds, convert them into, by filling them, we convert them into fields, or we bring them as a kind of human villages and real estates and other things. The thing is that these are the kinds of habitats. I would say that community, as a person who live in cities and the so-called civilized society, we should not put the burden on the community, but we also have to share the responsibility with the indigenous community in such a way that those habitats where the kind of wild varieties, crop wild relative plants are there, because in situ conservation is very important. In situ conservation, just we cannot go and put a placard saying that wild plant habitat, this is where the wild rice are there, we have to protect. There, our duty don't stop. So how do we protect it? So we have to, of course, community have to get to know this is a habitat for the crop wild relative, that is rice. And we also have to tell them that how it is important for the other paddy fields to be genetically diverse. We all talked about some years back, we talked about GMO crops, genetically modified crops. So we know that how the crop wild relatives are really contributing in bolstering the genetic diversity of cultivated plants. So that way, I would say to conserve this kind of crop wild relatives, we have to go for smaller scale community participated conservation areas, more of microcosms that has to be conserved. And then they will be act as a kind of stepping stone across the human dominated landscape, and that will go and merge with the protected areas surrounding this human dominated landscape. I hope I have answered the question. That the answer, simple one sentence answer is that linking protected areas through this micro conservation area where crop wild relatives and wild edible plants live.
Yeah, thank you, Dr. Ganeshan. And this Porteracia is actually in a mangrove area here.
Okay.
In the backwater. Anyway, next question from again Joel. Are you moving towards creating a genetic bank of these interesting cultivars?
Of course, our National Bureau of Plant Genetic Resources periodically collect crop wild relatives and wild edible plants and keep in their germplasm, that is gene bank. I think that if systematically these things are going on, I don't think that we need to have the kind of effort. But traditionally, we say that we have 500 mango varieties and several hundred paddies, several types of garcinias. So these are all the efforts of our ancestors, our grandparents. So they know the value of this kind of wild plants and their contribution and their diversity of taste, diversity of diverse culinary preparation for which this diverse fruits, leaves, and other parts of the plants are very much useful. And we also know that tribals, I'm sorry to use the word tribal, indigenous community, still they celebrate various festivals, especially recently during Sankranti time, they might have celebrated tuber festival. That is, we call, in Kannada, we call it as Ganeso Haba. So these are all the kind of celebrations and practices and tying or making ourselves intricate with the cultural practices are some of the ways through which we'll be able to still conserve the germplasm or genetic diversity of this kind of crop wild relatives and wild edible plants. In addition to passing the bucks to National Bureau of Plant Genetic Resources, forest departments, and other NGOs who are into conservation.
Sir, a couple of more things are there. This is from Dr. Vinayak Upadhyaya, Assistant Professor of University of Agricultural Sciences, Dharwad. Of course, he has also commented, I thank Dr. Ganeshan for a very nice and informative talk. Sir, can you enlighten us regarding wild edible mushrooms and conservation strategies?
Okay. Yeah, it's a very, very important and pertinent question because there was a question by Dr. Kerker about ferns and of course mushrooms. And especially in South India, we see only two types of mushrooms. Whereas if you go to Northeast or any other part of Southeast Asia, including China, we see diverse mushrooms are part of their day-to-day culinary preparations. Of course, at the global level, especially many European countries are making special efforts to protect and conserve the in situ conservation efforts regarding the mushrooms. So in India, we are not doing other than collecting it from the wilderness. I think we have to seriously think about how we can still domesticate few more wild edible mushrooms, and what could be the other alternative to produce more in the more in our human-dominated landscape. But definitely mushrooms are very, very important, and at present they are not at all taking the center stage, neither as a wild edible plant nor as a species to be conserved.
Yeah. And sir, as an information, our former colleague, Dr. Kamath, was trying his level best to bring certain wild mushrooms into cultivation. And of course, to what extent you could succeed in that, we are yet to know. But he was trying a lot. Sir, one more from, I think we will take only this one and one more question. Dr. Arshala was raising her hand. This is from Akshatra Fernandez. I must thank her first. She is the rapporteur. Rather than doing her job, I think she is asking some question. Anyway, Akshatra, I will read it for sir. Sir, in Goa, earlier we used to eat echinoclova and paspalum, which you mentioned earlier. But now, due to new rice varieties, these plants are least known and are known weeds. Are they nutritionally important? And is there any scope to conserve these species?
Part of the one of the question also I answered that nutritionally they are very, very important. Important in the sense that they are in a very stressed condition. When generally those are all cultivated as a crop, more a dryland crop, just plow the field and then we sow the seeds and reap the grains from them, reap the cereals from them, or reap the millets from them. The thing is that compared to cultivated crops like paddy or wheat or any other cultivated cereal, these millets are very rich in various other nutrients which are very, very critical for our health, especially developing immunity, and also the fiber content in the millets are very, very important. But unfortunately, because of the same reason, processing to make it as a kind of grain for our cereal for our part is what really taking time. And I think with the world at which the speed of day-to-day life, many of us got addicted to using the rice or wheat and other grains, and then we hardly substitute, at least I'm using the word substitute, from the point of carbohydrate requirement, we can still think of using these millets. The thing is that unless otherwise we force ourselves in using it, we will not be able to talk about conservation. So when we talk to a human very, very honestly, they will say that if I'm not going to benefit out of a so-and-so species, why should I conserve it? Why should I bother about its habitat to be conserved? So that way I would use the same argument that unless otherwise, if we are not going to use, by using this, we are not going to make a huge demand on these cereals or the kind of millets, there won't be any farmer who will be interested to cultivate it. So I feel if we are, as a consumer, if we start consuming more and more millets, more and more wild edible plants, definitely that will be one of the important steps that we will feel why we have to conserve. For example, one of our colleagues now mentioned Puerto Rico. Again, Dr. Janardhanam gave me new information that it is a mangrove grass species that is wild relative of the rice. So if we are not thinking that the rice is an important, commercially important or important crop, why do we bother about conserving the crop while it is of the rice? And why we are making special effort to collect more and more crop oil relatives and then using them in crop improvement experiments. Nowadays we use a lot of genetic studies. Genetically we try to improve our crops. So the message I would like to give all of us, all of you, is that try to consume millets. This is not only going to improve your health, but also ultimately you are going to improve the conservation of crop oil relatives, especially the important millets and their crop oil relatives. Of course, our India is one of the cradle or the hotbed for domesticating many such grains. I hope I have answered your question.
Yeah, sir, thank you. I think there are no more questions unless someone comes out. But if there is nothing, I shall thank you, sir, for accepting our invitation, giving such a wonderful pan-India talk, in the sense examples from north, east, from dry land, from Western Ghats, from everywhere, from your personal experience, which is very hard. Most of the people, put little and take a lot from literature and do it. And this is based on the first-hand experience. And we are really thankful to you for accepting our invitation and for being with us and giving such a wonderful knowledge. Thank you, Dr. Ganesan. And I also thank Ms. Akshatra Fernandez, who was rapporteur. And I thank Dr. Siddhi Jalmi, organizing secretary, and Professor Krishnan, HOD and convener, for giving me this opportunity to chair this session. Thank you, sir.
Thank you. Thank you, Dr. Ganesan. Thank you. It's a nice, wonderful talk. Yeah. Ganesan.
Thank you, Dr. Krishnan.
Yeah, thank you. Thank you. Yeah, thank you.
So, shall I resign myself? Thank you.
Yeah. You can.
Thank you.
Thank you. Thank you very much. Bye.
Thank you. Bye.
So, Siddhi, tomorrow at 10 o'clock, I think.
Yes, so we can everyone meet tomorrow at 10 o'clock for the 6th session.
Yeah.
Yes. Thank you so much.
Yeah, thank you.
Thank you,
everyone.
It went on well, first day.
Yeah, thank you.
You can cut your YouTube connection.
Yes. Dr. Siddhi, yeah, please do that.
Yes.